Hello, good afternoon, everyone. Get started in a little bit. Just waiting for everyone to join in.
Good afternoon, Coach Joe.
Okay, so let's get started. Super excited today. Our thesis defense for our 2025 spring season. I want to thank everyone for being here. This is the format. I will call your name, and you will need to turn on your video, give your presentation, and after that, you'll have to field two questions on the chat. So everybody, you've got to stay till the end of the presentation, ask two questions in the chat, and let's get started. Okay, so let me go with Tanishka. Are you here, Tanishka?
Yeah.
Okay, you want to turn on your video? Yeah, I see you there. Let me pin you. Um, Yeah. So please go ahead. The format is you finish your presentation, then Professor Barkey will ask you a question, then Professor Jen will ask you a question, and then you can answer them, and then you will have to answer the questions that come in the Zoom chat. Got it? So let's get
Okay.
started.
Okay. There we go. Is this working?
Yes.
Okay.
Yes, I can see your presentation.
Okay. Yes, sir. Yeah. So I was setting out to study the impact of genetic variability and environmental factors on schizophrenic development. My intro, obviously, topic is investigating in schizophrenia, and a couple of questions that I sought out to discover were first the genetic impacts of schizophrenia, because it's widely known that schizophrenia is pretty much genetic. So, just to study the different genes, that was one of the questions. The other one were specifically the environmental impacts, how you're raised, what your parents are like, just overall living conditions. And then cognitive effects were your after effects for schizophrenia. So then the objectives were simply just to understand all these different sorts of impacts on the development of schizophrenia. And over here is the picture of the specific symptoms of schizophrenia that we perceive as schizophrenia. So like delusions, hallucinations, like the flat effect where you just have no facial expressions, and then just disorganized thinking patterns. All these sorts of things is how we truly see schizophrenia as. So this is just a little like picture. Then the literary review, so like some of the sources that I used were for Cambridge University, for example, the specific sorts of genes, like the scientific names of them, in order to find like what they do, their genetic molecular component as to how they are with like the DNA. Then I also use sources from Mayo Clinic for the specific environmental triggers. So, like we stated before, just the how you're grown, how you're raised as, all those sorts of things. And then the limitations are obviously that schizophrenia is still very much undiscovered. There's not a lot known about it. So the sources that I had to use were obviously secondary sources. And so I can't do my own studies or conduct research based on that. And then my methodology and results were secondary sources, case studies, like twin studies, for example, to prove and show the genetic component of schizophrenia, and just overall different articles on different sorts of behaviors and patterns. And the results were, although schizophrenia is definitely a genetic component that was proven, it's not valid to say that that's the only thing, because you could still have schizophrenia, like the genetic predisposed to schizophrenia, but if it's never triggered by an environmental factor, there's a pretty good chance that you might not even see in your life anyways. And then the conclusion was just simple, how the genetics are the crucial role factor that play in the development of schizophrenia, how the environmental is definitely a main trigger point in order to show the activation of the disease, and then the cognitive effects are just our primary way of perceiving what schizophrenia is. And hopefully the contributions that I've made is opening a new door for research and further discovery into the study of schizophrenia and bigger overall impacts in understanding the disorder. Yeah, that was it.
Okay, you want to share your experiences with Gifted Gabber?
Yeah, so this really helped me really dive deeper into schizophrenia. I've always been interested in it, so this just helped me unlock it more and like actually learn more about schizophrenia. And I think the professors have been really helpful in giving good information about the DNA, helix structures, all these different genes, as well as overall like proofreading it, making sure that the research paper obviously like turns out good and it's just effective for all audiences to be able to understand, not just researchers or scientists, but for the general public.
Wonderful, wonderful. Professor Varkey, a question for Tanishka.
Tanushka, good presentation. Just wanted to ask a single question that is, do you know how like they correlate a particular gene with schizophrenia?
Yeah, so this connects to the cognitive impact actually. So the genes obviously that are part of your helix DNA structure, these are just there. There's certain mutations in the genes, and then these different types of mutations are actually what affect the different areas of our brain. So like the cerebellum is one, amygdala is other. These just genes are connecting to these pathways in your brain, and then how we see schizophrenia as it's activated through the specific regions that these genes affect.
Do you know, by the way, like how they actually pick a gene? Like how do they actually correlate one particular gene with schizophrenia?
Um, it was never stated in the articles. They just told me the specific gene patterns and like just how it affects schizophrenia, not how it happens really. The mutations are just predisposed, I'm pretty sure for most of them. So it's just like that.
Okay, maybe like that's something could, I'll tell all the students anyway, that if you keep on working on the same article, you can improve it and add more information. This could be something that you might want to consider adding on to your already, like the presentation that you already have or your research article that you prepared. Good job.
Wonderful, wonderful. Let's have a question from Professor Tuban. Professor Jen, you want to ask Tanishka a question?
Yeah, before that, I'd like to say that you have done a fantastic job presenting your research, Tanishka. So congratulations for that. I just want to ask, like, what would you say is the most significant contribution your research makes to the existing literature?
So existing literature as in like what's already been published about schizophrenia.
You're correct.
Okay, so a lot of the articles that I was reading through, especially, there was no like it was all very focused on one area. So either they just focused on genetics or they just focused on the environmental. There was never a total complete case study of everything. And like I said before, I feel like schizophrenia, although it is mainly identified as a genetic disorder, it's important to consider that environmental factors really do have a bigger impact as well. So I just felt like all the articles don't really talk too much. They just like focus on their individual prime topic. So I think that my article is just easier for people to overall like not, in the sense, like be more accurate when they define what schizophrenia is, not just correlate it with one or the other, but both.
That's all. Thank you so much for that.
Okay, great job, Tanishka. And two questions for Tanishka on the chat. And Tanishka, you have to stay till the end of everybody's presentation and answer your questions, and of course, ask each other. So we want you to learn not just from your topic, but from your friends as well. And we want to build a tribe of students who depend on one another. So we want you to uplift each other. So please do not leave as soon as your presentation is done. stay till everybody is finished and ask 2 questions. Okay, great job, Tanishka. 2 questions for Tanishka's topic, which is schizophrenia. Okay, so now let's go with Varsha. Varsha, are you ready?
Yeah, I'm ready.
Okay, I'm just going to, okay, go ahead, sweetheart.
Can you just give me 1 second?
No problem. You can just click the present button. Click on the present button right there and present. That's it.
Hello, everybody. My name is Varshamote. I'm a 10th grader at Mount Whitney High School, and thank you for coming to my thesis defense. My research paper was on evaluating the accuracy and effectiveness of wearable devices in predicting the episodes of bipolar disorder. So, through my thesis defense, I'll be going over the introduction, literature review, methodology, results, the discussion, the conclusion, and I'll be sharing some limitations that I've found, some recommendations that I suggest, and a thank you. So the focus of this research paper was to find the connection between how a patient sleeps and their overall circadian rhythm and how that can affect their mood shifts within their disorder. The objective of this research was to find the answer of whether sleep or wearable devices can accurately and effectively predict the mood episodes of bipolar patients. And the findings of this research can effectively personalize treatment and prevent patients from falling into relapses or hospitalizations, and it can overall affect how well a patient copes with their disorder. So throughout my research, I found 3 key findings. The first one was that disruptions in sleep can cause a lot of impacts on the symptoms. So disturbed sleep can generally create more severity in their symptoms, and it can also make their symptoms much take place more longer for more duration. The second key finding was that sleep studies and devices are actually very effective in predicting each episode and predicting when episodes might take place, which can also give patients and both also their healthcare providers a heads up on what their next step should be. The third key finding is also that these wearable devices can also play a very big impact in treatments. And so patients and their providers can work together as a team between each appointment to make sure that the patient is safe between these visits to reduce complications in their disease or disorder. To give a little introduction and to clarify some of bipolar symptoms, bipolar is a mood disorder in which patients can experience emotional highs at a varying intensity. This includes manic disorders in which patients are very impulsive and very hyperactive. It can also include episodes of emotional lows, which can be described as depression, in which patients are less active and overall they're not doing very well mentally. This disorder affects primarily the prefrontal cortex, the hippocampus, and the gray matter of the brain. And what it does is it shrinks or decreases the volume of each part of the brain, which also leads to less function in the brain. It also affects the dopamine, serotonin, and norepinephrine hormones in that when someone is going through their manic episode, their dopamine hormones are more active than the average person's. And when someone is going through a depressive episode, their serotonin hormones are working less efficiently than the average person's. And here are some images that can more explain this. Throughout my research, I also found more on how mood disturbances can be linked to how a patient sleeps, and the polysomnography explains a more standard effect of how when you sleep, certain numbers are given to each level of your sleep, and if you sleep more or if you sleep less, that all gets calculated into your overall sleep study, and that can be used to help predict your sleep or predict your episodes. And these wearable devices and smart applications can also um, pick up on other aspects, which is also your activity level and your heart rate, which is more holistic and can better predict your overall episode. The methodology, I also collected a lot of journals and data from PubMed, which were all primary studies, and they all proved that these wearable devices can accurately predict the coming episodes of each patient. And with this, it also improves a lot of clinical settings, and it improves the treatment of a lot of patients. This image shows how they used a standard equation for each sleep study and how they are able to pick up data from the sleep and being and able to accurately predict the data and accurately find the data. Basically, that the devices are able to capture the data accurately. This also explains the same thing, but more on how each device, like different devices, can capture more data more efficiently. So they're basically testing first, can the devices actually capture data instead of just can they predict it? We need to make sure that they can capture the data accurately first. This graph also shows that with the data that the devices have collected, can they also accurately predict the moods? And it's been shown that it's been up to 80% or more percent accurate, which means that it's a very efficient way of finding out which episodes come for each patient based on their sleep. A lot of these studies also said that if you have regular feedback for patients and if you give them ways to improve their sleep, ways to live a more healthier lifestyle, that actually decreases their symptoms and actually makes them have a more healthier lifestyle. And also the other information that we found were that these wearable devices, such as the Oura Ring, Fitbit, and Apple Watch, are actually up to 90% accurate in measuring the sleep duration, sleep efficiency, and overall just predicting the mood based on their data. And we found that Fitbits are actually very accurate in predicting these mood episodes because they are able to analyze a lot more data and they're able to capture this data and send it off to an algorithm, which is able to very accurately predict this data. We also found that because these wearable devices are very accurate, they can also help in a lot of clinical settings and help in treatment, which will further help the patient later on in their treatment. To conclude, the wearable devices are very accurate in monitoring how a patient sleeps and also how that can be used to predict their mood episodes, which can advance treatment tremendously and give patients a more easier time coping with their symptoms and more easier time dealing with the struggles that bipolar offers for patients. Each literature review also summarized and synthesized the past information that other researchers have collected. And through this research, I've also identified gaps within the current knowledge, which through further research can be discovered or applied more intensity on. Further research, I believe, should include larger populations within their research because previous research only included it on more smaller populations. And because of this, we might not be able to gather the whole holistic evidence because it's only a small sample of the research population. So some key limitations I've came up with is that, of course, a larger population would need to be tested on. Also, that these devices need to be able to capture the whole situation, and oftentimes they can't because these devices pick up on activity levels. And so if a patient is just lying there on their bed and they're not actually sleeping, but they're very sedentary and they're not moving, the devices can actually capture the data as if they're sleeping, and that data might not be accurate because they weren't actually sleeping at that point. So maybe further research should account for this. Another limitation I found was that a lot of patients might not actually agree to their data being tracked on through an algorithm, and so it could also lead to a lot of misuse of their data and a lot of privacy issues, and so patients might not want to use these devices because of that. And another limitation would also be that these devices can also be very expensive in the current world and the current setting. And so that can make it very inaccessible for a lot of patients to buy because of financial costs or even technical constraints. So with this research, I would recommend that, of course, researchers conduct research on a larger population, but also to create a standard system in which even clinics can use the data and use that for further treatment options, and also to come up with treatment options for other patients. They should also include a more ethical and legal consideration for the patient's safety. And so patients don't have to worry or stress about their personal information being used in a abusive or bad way. Thank you. My name is Varsha again, and I'm from Mount Whitney. And thank you for your time.
You want to share your experience with Gifted Gabber?
Yeah, so at Gifted Gabber, I would just like to say thank you, Dr. Varkey, for all the information that you've taught me and many other students. I found your guidance and support to be very helpful, and I would just like to thank you. Also, Miss Tubin, I would like to say that your guidance and support was extremely supporting of me, and it really helped me go through this entire process with a lot of ideas. And thank you for your suggestions on helping my paper become better. I also want to say thank you to all my other mentors because you really helped this process become more doable for me. Um, this um, this also helps me with learning more on bipolar. I've always found bipolar more interesting, and with this literature review process, I've found more knowledge that I don't think I would have learned if I hadn't gone through this process. And so I would just like to be grateful for learning about this. And I would also like to thank Coach Joe for what you've done. Um, I also gained a lot of experience in this. I learned how to create a literature review, and I learned how to actually go through papers and learn a lot of concepts and information. And I feel the most thrilling part of this situation, or the most thrilling part of this overall process, was capturing a lot of the data because I found learning all this was actually really interesting. And I feel that later on, I'll be more open to learning more information on bipolar.
Okay, wonderful. A question from Professor Varkey. Great topic, a very relevant one. Thank you for your in-depth presentation, Professor Varkey.
Thank you, Varsha, for your kind words and also excellent presentation. I can see that you put a lot of effort in preparing this and also read quite a number of articles. Uh, uh, just like one question for you that have you, like when you were researching or like going through the different articles, did you find an example where they used some kind of intervention based upon this wearable device? For example, if like they see that okay, there's a pattern that based upon sleep, you are going to have a mood disorder or manic episode. Even to prevent that, is there some intervention that they suggested and that it worked for them?
So, from the research that I've read through, they also said that based on the patterns of patients' sleep and like their overall circadian rhythm, they would provide feedback like sleep hygiene or that patients should sleep from a certain time or overall how a patient can improve their sleep, which has also
Mhm.
proved that patients have less severity in their symptoms and also their symptoms happen for a much shorter amount of time and at less frequencies.
Very good.
Great job. Question from Professor Chuban.
Yeah, first of all, I just want to say excellent work on your presentation, Varsha. You have clearly put a lot of thought into your research, very obvious. And I really appreciate how well you were able to convey your findings, especially mentioning the limitations. That's very, very impressive. Now, I just want to ask one question like, what new areas of study do you think your research opens up after after the findings and presentation and after producing the research paper?
I think with my research, it also opened up more on how patients can um almost help themselves more personally, and it can include more of like an electronic, more technological aspect in treatment where it's not just, oh, let's have a talk and then I give you a medication. It's also included more on their holistic life, how they can be more active in their life and have an overall better lifestyle because your mental health doesn't just come from an hour or 45-minute session, it comes from your overall day-to-day life. And I think this opens up how your nutrition, your sleep, your activity level, just what you do throughout the day, your overall health can come into play with your mental health.
Thank you so much.
Yeah, I mean, this is very, very good. I feel like dealing with mental health illness is more critical because the symptoms are not very clear, right? It's like it can easily be brushed off, right? As against your having diabetes or something where it's very clear, right? Your monitor and everything, but mental health is something very, very subtle that gets can slip through the cracks. And I feel like constant monitoring and whatever research you've done is going to be extremely impactful. So great job. Two questions for Varsha. I don't see any questions for Tanishka. Please, two questions for Tanishka. Your research performance is also based on the quality of the questions that you ask. I'm a Tony Robbins life coach, and he often says that the quality of your life is dependent on the quality of the questions you ask. So we would like to see that. We want you to be curious about everything, including others' research as well. So let's get going. Two questions for Varsha, two questions for Tanishka. Okay, so let's go with Shrika. Shrika, are you ready? You want to go?
Yes, I'm ready. Give me a minute.
Sure. Next up is Sri Harish. Shrika, if you need a minute, we can go and come back. Got it?
Okay, yeah, that's fine.
Okay, Sri Harish, do you want to go ahead?
Yeah, sure. Let me pull it up. Just one sec.
No problem. You want to turn on your video?
Oh yeah, sorry about that.
That's okay.
Can you see my screen now?
Yes, but we can't see you.
Oh, 1 second.
Okay, there you go.
Better?
Good
All right,
stuff.
great.
Yes.
All right. Good afternoon, everyone. My name is Sri Harish Kannan. I'm a junior at University Laboratory High School in Urbana, Illinois. And today I'm going to be doing my thesis defense on the effect of photobiomodulation on Parkinson's disease. One sec. Okay. So just for like a brief introduction about what my topic is, I'm going to be investigating how light therapy, specifically photobiomodulation, which is a form of low-level light therapy, can impact research in symptom alleviation as well as future prevention of Parkinson's disease. So I find this topic really important because Parkinson's disease is the second most common neurodegenerative disorder, and it primarily affects motor function and significantly diminishes the quality of life of its patients. Some research questions that stemmed from my initial topic were that what wavelengths of light and photobiomodulation are most effective, and what treatment durations are most effective as well. And as well as I wanted to learn more about what stage of Parkinson's progression does photobiomodulation actually impact with and help with. And then my final objective of this whole paper, of this whole literature review, is to determine, is photobiomodulation a viable treatment as a complementary treatment, as well as like pharmaceutical treatments like levodopa, which is a precursor to dopamine, and MAOB inhibitors. So in the literature review, one key pillar is the fact that mitochondrial pathways are involved in Parkinson's disease pathogenesis. They are especially critical for the dopaminergic neurons, which are 4.5 meters in axonal length and have 2.5 million synapses. And these means that these neurons are highly active, and they require a high metabolism. Hence, they require efficient mitochondria. As you can see here in the diagram D, we have in this cell, we have a protein called asynuclein. So as you can see here, it's aggregating, and this is what happens in Parkinson's disease. The asynuclein protein starts to aggregate in the cell, which blocks off the neurons from other neurons, so it can't transmit chemicals like dopamine, which are required for motor function. Normally, in a cell that isn't affected by Parkinson's, autophagy, which is on the right, will occur when the mitochondria is efficient and adequate ATP is produced. However, if the mitochondria is inefficient by inhibitors, the adequate ATP will not be produced, autophagy will not occur, asynuclein aggregation will happen, and Parkinson's disease symptoms onset onto patients. The second key pillar in my literature review background is the fact that cytochrome C, there's in the mitochondria, there's something called the electron transport chain. And the electron transport chain is fundamental for what produces ATP in the mitochondria. Specifically, what's affected in Parkinson's is cytochrome C oxidase, which is the fourth step, right in the green. It's the fourth step in ATP. So in ATP production. So what happens here pretty much is that cytochrome C oxidase is giving out electrons, but slowly you see there are these nitric oxide molecules. They start to inhibit the cytochrome C oxidase when this asynuclein protein, which we saw in our last slide, starts to block off the neuron, because the neuron starts to think, oh, it's in trouble, it needs to do something to fight it. So the nitric oxide starts to inhibit that fourth cytochrome C oxidase. But what occurs is when we put light therapy or like photobiomodulation, what occurs is that it dissociates the inhibitory nitric oxide when it absorbs photons from light therapy, because the nitric oxide basically binds to the photons and then it goes outside the cell. And this triggers a cascade of events, which will allow for efficient mitochondrial functioning and the regeneration of ATP, and allows it to produce chemicals like dopamine, which are important for our motor function. So in my actual research paper, I analyzed secondary sources through qualitative results through sources like PubMed, where they described how Parkinson's disease the onset of Parkinson's disease, the effects of the Parkinson's disease, how it affects the body, as well as looking at primary trials, also on PubMed and the National Health Institute, where they had specific tests on specifically biomodulation, different types of wavelengths, and like how long they would treat them for. And those were my two main sources that allowed me to make my key findings in my literature review, which is that biomodulation is an effective way to fight Parkinson's early on. If you can, this was proven in animal clinical trials. And what this means pretty much is that if we can determine Parkinson's from an early stage, so we can see it happening, we can do photobiomodulation through intracranial method. And what that'll allow is for basically the mitochondria to be reproducing, basically to regenerate the full function of the mitochondria. The second thing I found to answer my question about wavelengths and which is most effective is that a wavelength from 800 to 850 nanometers is most effective. So the reason for this is because without damaging the human body, our bodies can only sustain a wavelength from 600 to 900 nanometers. But at the same time, our brain is surrounded by a thick skull. And in Parkinson's, it specifically affects the substantia nigra, which is in the midbrain. So to be able to effectively reach the substantia nigra, we have to have as high as a wavelength as possible. But what these two findings that I found really what I wanted to think and what I talked about in my research paper is that there needs to be more research done on this topic. It's something that can have a solution. It's proven through animals that it's possible. We can actually fight Parkinson's with this. However, we need to start doing human trials and actually observing in our own species how this works. But some limitations, when we talk about going to human trials, there's many limitations to it as well. One limitation is that there are many different pathways for how Parkinson's occurs. It's not just through mitochondrial dysfunction. There's also studies that show that there's something in molecules in the intestine which can cause Parkinson's. And additionally, the other big problem is that currently there's only invasive techniques to get the light to the brain. On the right, for example, there is an intracranial technique, which basically means that a thick rod would be inserted through the brain to reach a substantia nigra to provide photobiomodulation. And the problem with this is that, one, it's invasive, it causes a lot of, and you need a big surgery for this to occur. And additionally, if an infection or a sepsis starts to occur, it's very hard to fight. It can be very harmful to the patient's life. So overall, what I emphasize in my paper is that we should do more research to find a safer and more efficient way to use photobiomodulation to fight Parkinson's, because it is possible. So finally, I just wanted to thank everyone at the team at Gifted Gabber for supporting me, especially Dr. Varkey for giving me great insights during the group sessions and the one-on-one sessions, as well as Coach Jen for checking in on me and making sure I was doing and helping me throughout this process, and even helping me after this process to make my research paper as good as possible. Thank you.
Wonderful. You can stop sharing. I'll have Professor Varkey ask you a question. Yep. Yes, Professor Varkey, a question for Sri Harish.
Excellent presentation, Sri Harish, and very unique topic too. So just a couple of things for you. So one, do you know that if this particular method has been used for other neurodegenerative diseases also? And just wanted to ask another question. So you mentioned that they put like a big rod into the brain to cause substantia nigra, So is there any alternative that you can think of?
I'm going to answer your second question first, Dr. Varkey. So the substantia nigra, it's located in the midbrain. So what that means is that it's most easily accessible by the sphenoid bone, which is like right here, and then the occipital bone, which is in the back of the skull. So what that makes me think is that there could either be I think the most effective way, that if we're not just like sticking through the brain from the frontal, would be going through the nasal passage to the substantia nigra, because there is a clear-cut path through there. I think that would be the best way to interact with that situation. For the other question, I've seen that they've used photobiomodulation on Marfan syndrome, which is when you also another genetic disorder where you have it's a genetic disorder, but basically you can't control your arms and legs, and leads to paralysis at old age. So I've seen that they've used this, and they've actually tried it on humans, but it's only had varying levels of success. The main problem with I found with those studies is that they use sample sizes of 10 people, which I understand because not many people are willing to test their life for a half chance of surviving. But I feel like if they were able to get bigger sample sizes of a couple hundred people, we could act better idea of is this actually going to work? Because in study with 10 people, only about like 4 of them said that it was actually like helpful, like they were able to move more and come back to normal life to a certain degree, of course. And 3 of them were actually said it made it worse, and the other 3 said there was no effect at all. So I just feel like if there is more actual research into the effects of photobiomodulation in humans, we could actually see if there's a better effect on not only Parkinson's but other diseases like Marfan too.
Excellent answer. Good job, Harish.
Thank you.
Excellent. Let's have a question from Professor Jen.
So great job on your presentation, Shri Harish. So it's very evident that you have worked hard to produce this paper. That's very, you know, impressive and fascinating to hear about your approach and the findings. You also mentioned that there might be further research to be done based on this study. So I would like to ask, what recommendations would you make for researchers interested in exploring this topic further? Like, what should they consider? And are there any particular areas that they should focus on and that you haven't mentioned or you haven't included in your paper?
No, that's a great question. Thank you. So, that I haven't mentioned, I'm just going to talk about something I mentioned already first. So, obviously, the first step, in my opinion, would be to go through to human trials. But I've already talked a little bit about that, so let's talk about something different.
Yeah.
I think that another avenue that you could take through this photobiomodulation, not only looking at just human but where exactly will it be where exactly in the mitochondrial process where will photobiomodulation be the like most effective? I know that I talked about in my presentation that if you do it early enough, it'll work, but if it's too far gone, it might not. But that's honestly the reason I didn't really give a clear-cut answer to that is because it's a really big gray area in terms of research currently. There isn't much there aren't any articles on fully understanding how Parkinson's develops and finding an accurate process to determine how far how far are patients in terms of the process of Parkinson's. Like in cancer, they have stage 1 through 4. Parkinson's, it's not very clear cut situation like that. So I think another avenue of this research should be how can we determine how far you're along in Parkinson's, and that way we can even create prevention treatments rather than just see what happens this far late into your life.
Good job.
Sounds
Good job.
good. Yeah, great job. Thank you for presenting, Sri Harish. I just want to let everybody know that you have to ask 2 questions. I don't see any questions for Tanishka and Varsha. So yeah, please start asking questions. I'm going to start reading out everybody's names at this point. Aishwarya, Ajay, yeah, Aishwarya, Ajay, we need questions to make sure you are listening to your compatriots' research. Aditi, Arnav, Amol, I mean, yeah, please start asking the questions. And of course, you need your pesky certificates. We need to see those questions going. So let's get ahead. A very good presentation, Sri Harish. Very nice rebuttal and answers as well. So let's go to Shrika. Shrika, you ready?
Yeah, I'm ready.
Yeah, you want to turn on your video?
Yeah, can you see me?
Yeah, okay, great. Let me just pin you. Yeah, go ahead.
Okay. Hi, everyone. My name is Shrika Manabolu, and I'm a junior at Rockridge High School. And today, my topic is music therapy's neuroprotective role in Alzheimer's disease. And before I go further, I just want to say the purple color that I use for this slide presentation represents the Alzheimer's Association. Okay, so think about music, not just something that we listen to, but something that could actually help our brains. So I actually just got back from a piano recital this morning, and it made me realize the importance of music and how powerful it really is for our brains. So that's why I want to dive into how music therapy can do just that for Alzheimer's patients, potentially helping to save their memories and cognitive function by giving their brains a chance to reconnect. So the title of my research paper is The Neuroprotective Effects of Music Therapy in Alzheimer's Disease and Its Role in Neuroplasticity and Cognitive Preservation. So I understand there's a few big words in my research title, so I'm going to define them now. So neuroprotective effects is the way something can protect the brain from damage, keeping it healthy and preventing diseases like Alzheimer's from causing them. And then neuroplasticity refers to the brain's ability to change and adapt. It means that the brain can make new connections and heal itself, even when some of the parts are damaged. And then cognitive preservation refers to keeping your thinking, memory, and decision-making skills sharp for as long as possible, even if you age or face challenges like Alzheimer's disease. So moving on to my introduction. So Alzheimer's disease is a progressive neurodegenerative condition, and it primarily affects memory, causing confusion and cognitive impairment. And Alzheimer's disease is characterized by the accumulation of beta amyloid plaques and tau tangles in the brain, which can disrupt the neural communication, leading to widespread brain damage. And then currently, the available pharmacological treatments primarily focus on managing the symptoms, but they don't halt the progression of the disease, which is why people resort to music therapy to help. And then here I have a picture of 3 brain scans. So as we can see in these brain scans, it shows the progression of Alzheimer's disease. So on the left, we have a normal brain scan and it shows balanced activity. And then as we move forward to the middle, we have the mild cognitive impairment in the middle. And we notice a slight decline in brain function. And then finally, on the right, in the brain of an individual with Alzheimer's disease, there is significant atrophy and diminished activity, especially in areas critical for memory and cognitive function. So now I want to go into my literature review, and this is some of the findings from the existing research on music therapy for Alzheimer's disease. So first, music therapy has shown promise in stimulating new neural connections. And then we also have active versus passive therapy forms. And then studies show that it also engages various brain regions, including the auditory, motor, prefrontal cortex, and hippocampus. And then this type of therapy has also been found to improve memory, fluency, attention, and executive function. And it's also been shown to enhance mood, reduce agitation, and promote social interactions while increasing important neurotransmitters like dopamine, serotonin, and oxytocin. So now moving on to my methodology. So I first conducted a literature review to explore the effects of music therapy on Alzheimer's disease. And then I also used neuroimaging techniques, as you saw on the last slide, including fMRI and PET scans to study the brain activity before and after music therapy. And then I also compared the effects of music therapy with other auditory stimuli, such as familiar voices or standard care, to determine the most effective intervention for improving cognitive function. And then, as always, I also use Google Scholar and PubMed for my research. So the results of my paper. So it indicated like significant improvements in cognitive function, especially in verbal fluency and memory. And then the neuroimaging scans also showed increased brain activity after the music therapy. And then the therapy was also linked to reduce agitation, anxiety, and depression, improving the mood of and behavior of the patients. And then it also enhanced social interaction as patients were more engaged during sessions. And then long term, it seemed to sustain neural connectivity, which could potentially help slow the progression of the disease. And then my conclusion slide. So in conclusion, music therapy has demonstrated neuroprotective effects by enhancing neuroplasticity and preserving cognitive function. And it's also been shown to improve emotional well-being, making it a promising non-pharmacological intervention for Alzheimer's disease. And then importantly, music therapy could also complement existing treatments, and it can help improve the quality of life for patients living with Alzheimer's. And then now going into my experience and acknowledgements. First, I would like to thank the entire Gifted Gabber program for giving me this opportunity to be here right now. And then specifically, I'd like to thank Coach Joe, Professor Barkley, and Coach Jen for helping me with my research journey. Dr. Barkley taught me a lot about the field of neuroscience, and my creative writing professor, Coach Jen, taught me a lot about how to organize my paper and gave me insight on how I should structure each paragraph. And so I'm very thankful to the entire team. And yeah, thank you.
Excellent. You want to stop sharing so that I can get Professor Varkey? Professor Varkey, a question for Shrika.
Good job, Shrika, for the presentation that you just had. And like, I have a question for you. When you looked into the effect of music, did you find any specific areas of brain that are triggered when you listen to music? And is there in particular music better than others in promoting this cognitive improvement or even like neuroplasticity?
That's a really good question. So I mentioned the types of brain, like the parts of the brain in my presentation. And then, so I've mainly found that it's mainly like the prefrontal cortex. And because that's like the main part for memory. And then it's also like all the lobes. So like the frontal, parietal, temporal, and occipital lobe. And then for the question about like what types of music, I found classical music can really help the brain with memory, because I know playing piano, I researched into that. And it's mainly like playing classical music for patients, because Alzheimer's like usually affects people like of elderly age. So I found classical music was very helpful.
Professor Varkey, we can't hear you a bit. Your volume's a bit low.
Can you hear me now?
Yes, now yes.
Okay, yeah, no, I think Shrika, good job, good answer.
Yeah. Yeah, Shrika, very interesting fact about the classical music. Wonderful. Thank you. A question from Professor Jen.
Yeah, thank you for your presentation, Shrika. It was very clear, focused, and engaging as well. You told us about the color purple in other image that represents something meaningful. And your research is of very high standard, because I can tell that you have invested a lot of time into both your research and the way you produce and interpret the findings and the literature. Now, my question is, what what were some of the challenges you face when synthesizing literature and even writing your paper, and how did you address them?
Okay, so I found like a lot of challenges, because obviously neuroscience, it's very complicated, and especially like dealing with memory and cognitive impairment, there's a lot of challenges you can find and a lot of vocabulary terms that you don't know. So my main thing was just researching, because I couldn't figure out like some of the vocab terms. It was very complicated, like the beta amyloid and like the other vocab terms. So I think that was the most challenging, like trying to decipher what all those mean and how they all relate to each other.
Excellent.
Thank you for that.
Thank you so much, Shrika. Very interesting topic. Everyone, 2 questions for everyone here. Let's go with Soham. Soham, you there? Wasn't it Soham who requested me? I'm sorry. You were the one who requested, right?
Yeah, that's me.
Okay.
Can you hear me?
You want to turn on your video, honey?
Yeah, sure.
Okay, great. We see you there. Wonderful. Yeah, go ahead, Soham.
Okay, let me just share my screen. Okay, can you see my screen?
Yes, you want to click the slideshow? The slideshow?
I
Yeah.
did. Can you see it?
Yes, I can see it. Go ahead.
And can you hear me well?
Yes, perfect.
Okay. So hi, my name is Salam Balor. I'm currently an 11th grader, a junior at Ravenscroft School, and my research was investigating the impact of stress, anxiety, and fear on cognitive function, decision making, and neuroplasticity. So first of all, what are the major issues? Stress, anxiety, and fear are on the rise, not just in America, but around the world. The global population, over half of the global population reports increasing levels of emotional stress since 2007. And on Gen Z feels more stress on average. Rating is 6.1 out of 10, which is a zero point 0.5 increase from millennials, a zero point 0.9 increase from Gen X, and a 2-point increase from boomers. While that initial 0.5 increase may not seem like much, a large part of Gen Z has not entered the workforce yet, or still in college, or like us, are still in high school. So they haven't experienced those new stressors and new anxieties and new fears yet. Uh, there's a major significance to these rising levels. Each factor now becomes part of our daily lives. We experience it day in and day out, and it'll be short term as well as long term. And this is where decision making, cognitive function, and neuroplasticity come in. Decision making is a short term uh, process. We make a decision from the point we wake up to the point we go to sleep every single day. Cognitive function is sort of that middle ground. It occurs daily as well, but also memory and other things, especially long-term memory, occurs over a long period of time. And neuroplasticity is long-term. It's the ability of the brain's ability to adapt and reorganize itself, and that's a long-term process that occurs over your life. So I wanted to study the factors on, I want to study the variables on these three processes. So my literature review, I use most of my sources came from PubMed and NIH, and I used a few sources from JSTOR. My four major studies were these. Hewitt et al. In 2013 tested the impact of stress on cognitive function uh, in certain animals and linked it to E3 ligases and glutamate receptors. Santa Barbara et al. In 2024, they conducted a meta-analysis and what happens to memory over time. And Ting Ting Wu et al. And Sarmiento et al. Experimented and analyzed anxiety and stress impact on decision making. So my methodologies, I analyze different types of studies, meta-analysis, reviews, and experiments. And my research is primarily qualitative. My research findings found that chronic stress and anxiety are always negative in the long term. They hurt neuroplasticity and cognitive function. One way they do this is they reduce the level of brain-derived neurotrophic factor in the brain, which is this right here. Basically, what BDNF does is it promotes neuroplasticity and it promotes brain growth. But chronic stress and anxiety actually, when they're present, they take away the ability for BDNF to be transcribed. As such, neuroplasticity is harmed and cognitive function is harmed. Additionally, one of my other studies found that over a period of 28 years, anxiety leads to a 24% increase in dementia. And this correlated with other studies that said short-term memory is negatively impacted by chronic stress and anxiety. They compete for neural resources, and as such, it's hard. It gets harder and harder for short-term memories to be encoded into your brain. Acute stress and anxiety, however, it was found that they had mixed effects. Sometimes they could hurt, and other times they could actually help. Acute stress was found to increase glutamate levels, which can be neurotoxic. And it was also found to lower the levels of BDNF. But another study contrasted the original study, which stated that BDNF actually is regulated by acute stress and anxiety, and it's through glucocorticoid receptors. Acute stress can actually increase neuroplasticity and in turn like increase the ability of the brain to adapt. Anxiety was also found to, while acute anxiety was additionally found to hurt short-term encoding, it was found that it can actually improve your long-term memory facilitation. As such, both of them can help. For decision making, it was hard to find if there was a significant impact stress, fear, and anxiety had on the process because there were a lot of other variables that play roles in decision making. And it was modeled by one of my studies where a lot of these variables here that you can see, sex, age, culture, personality traits, and background, they all play a role in decision making. The type of stressor also plays a role in decision making, especially like how like the durability of the stressor, the type of the stressor, and how intense the stressor is. And the type of decision being made also plays a role. Is it a risky decision? Is it a decision that doesn't have much of an impact on the rest of your day, or is it a decision that won't have an impact or in the rest of your life, or will have an impact in the rest of your life? So it was found that it's too variable, and there wasn't a direct impact that stress, anxiety, and fear had on decision making. So my conclusion and limitations, anxiety, stress, and fear in small doses has a mixed impact. It can either better or worsen cognitive function and neuroplasticity. Decision making is an outlier. As I mentioned earlier, decision making, we don't know if there is a direct impact on decision making that stress has, that anxiety has, that fear has. However, when chronic anxiety, stress, they are always negative. They don't benefit. They always hurt neuroplasticity. They always hurt cognitive function. They increase chances of dementia. They take away neural resources that could be used for improving cognitive function and improving neuroplasticity. And my limitations were there's a lack of research, especially a recent research on the topic. A majority of my studies were found in the mid 2010s, and only a few were published recently in the past 3 years. And there's also a lot of ethical concerns because while they can test on animals, they can't test on humans because you can't incite anxiety or chronic anxiety and chronic stress into humans. Those are massive ethical problems and ethical dilemmas. And it's also difficult to study fear because fear in these studies, fear was associated with anxiety and it was associated with stress, but it wasn't isolated as a variable by itself. So that's another limitation. So my research experience, I like to thank Dr. Varkey with helping me find my topic of research, places to research, particularly PubMed and NIH, and explaining some of the terms in my research paper, which I didn't understand. I'd like to thank Professor Jen for guiding me through writing my research paper and teaching me how to go about my research. And I'd like to thank Gifted Gabber for giving me the opportunity to research this unique topic while enjoying it along the way. Thank you.
Excellent presentation, Soham. You want to stop sharing so that I can get
Yeah.
Professor Varkey? Yeah.
Very good job, very focused presentation. And just one question for you. Do you think like if we increase the amount of BDNF, it can somehow restore neuroplasticity or even like neuronal growth?
I think that like you're saying like inject BDNF into the system.
Or use some medications which can increase the production of BDNF?
Like I think in short term it could definitely improve BDNF levels, but like I said earlier, neuroplasticity is a long-term process. So chronic stress and chronic anxiety, as they continue on over and over, if you have to keep taking those medications, and again, we don't know the after effects of those medications at all. So we don't know like if we do like inject BDNF or try and supplement BDNF, we don't know like what it'll happen to other parts of the body or other parts of the brain which do like use BDNF.
Yes, very good answer. So yeah, because you don't want to take something which can increase the activity all around the brain because you don't know what particular pathway you're going to trigger. Good job. Very
Thank
good.
you. Thank you.
Fantastic. I almost feel like I mean, I don't need therapy sessions. I just need to show up here. I do struggle with anxiety, depression, and all that, and I could just use all your studies here. So a question from Professor Jen.
Yeah, before that, excellent job on your presentation, Soham. Like you have clearly put a lot of work and your clear explanation of the research shows you have a deep understanding of the subject matter. It's very challenging, I can say that. Um, but I have one question, it's general. If you have more time or resources that you can use, how would you expand on this research? Or are there specific areas that you would like to explore?
I think one area that I would like to explore is like fear isolated alone as a variable, because in a lot of these studies, as I mentioned earlier, like fear is associated with anxiety, but not all anxiety does cause fear. So it's like trying to isolate fear, of course, is a lot, but if you have the right resources and the right amount of time as well, like the right period of time, like a few years or something, then it's very possible to isolate fear and then try and find its impact on these factors. So I think with the right resources, you could isolate fear, but with like how much time is limited for some of these researchers and some of these experiments, it'll take a while to like try and isolate fear alone as a variable, which is why it's a massive limitation in this study.
Excellent. If yeah, sorry. If at all you need some case study, I'm there. I'm going to volunteer for any of these studies that is there. Like I really think that fear aspect that you brought in is kind of very good. It really manifests the anxiety, and for me, a bit of meditation and working out and burning off, like in the gym, like really helps me. So
Yeah.
wonderful,
Thank you.
great topic. Keep going at it and keep me updated about how to deal with anxiety and everything. Okay, great
Of course.
job.
Okay.
Two
Thank
questions
you.
for Soham. Next, let's go with Ayan. Ayan Desai.
So
Ayan,
I'm
you here?
going to share my screen right now.
Okay, hold on a minute. Let me just add. Yeah, go ahead.
Okay, thank you. Okay, is
Yeah,
this okay?
we can see your screen. Yes.
All right, thank you. Okay, so hello, my name is Ayn Desai, and this is my thesis defense. A little bit about me is that I'm a freshman attending Yorba Linda High School in Yorba Linda, California. I love to play basketball and have been intrigued with the field of nutrition and how it can impact on sports performance. And this is why I wanted to combine all of these elements in my research paper, along with tying the topic back to neuroscience. So my topic is the role of macronutrients in neuroendurance during physical activity. So a table of contents, I'll first go into the introduction, then literature review, followed by the methodology, and then the conclusion. So this is an introduction. Athletes work hard to perform at their best and win competitions, and they often use nutrition to help them get there. So most people know that macronutrients such as carbs, proteins, and fat can help with physical endurance, but not many people know about something called neuroendurance, which is the brain's ability to stay focused and think clearly during prolonged or demanding activities. So in sports, being successful isn't just about how strong or fast you are, but it's also about staying sharp mentally. So in my research, I look at how the macronutrients we eat can affect the brain during exercise and help athletes perform better both physically and mentally. So this is my literature review. I focused on a few different articles for carbs, proteins, and fats, and I'll give a brief overview of them. So the first two are about carbs, and a study published in 2001 by Carlo and colleagues found that carbohydrate intake was able to enhance cognitive function. So they take trained athletes who drank a solution and then ran for 100 minutes, and then they tested the reaction time after the exercise. And they found that the athletes who drank a 5.5% concentrated solution containing carbs and electrolytes were able to have a significantly higher reaction time at the end compared to those who took a placebo solution. So they concluded that the runners who consumed carbs were able to improve their decision and reaction speed after the run compared to the placebo runners. And the second source was a study published by Dr. David Beller in the Gatorade Sports Science Institute, and they looked at the effect of carbohydrates in a sports setting now. And the research showed that consuming a 5 to 8% concentrated carb drink was able to improve performance, and it depended on the sport. So soccer players were able to that consume the carbs were able to have improved precision when dribbling, and they had a 6% concentrated carb and electrolyte drink was able to improve body motor skill performance. So, and also consuming carbs was able to give a lower perception of fatigue in players. So the overall finding in this article was that carbohydrate supplementation during exercise was able to support mental and physical aspects of sports performance. So my third article focuses on protein, and a study published in 2011 by Breen and colleagues looked at the effect of protein intake after exercise and how it affects muscle protein synthesis. So to do this, they took trained male cyclists for 90 minutes at approximately 77% of their peak oxygen uptake. So they would consume a carbohydrate drink by itself or a carbohydrate drink along with 20 grams of whey protein. and they looked at the myofibrillar and mitochondrial protein synthesis were measured, and they did this with muscle biopsies up to 4 hours post-exercise, and they found that the protein and carb groups had a higher myofibrillar protein synthesis, but it didn't really affect the mitochondrial synthesis, and the study showed that the protein intake along with the carbohydrate intake can improve muscle repair, and this can indirectly support cognitive endurance as it boosts recovery. And my fourth study focuses on fats. So a study published in 2023 by Ashton and colleagues looked at the MCT, which is medium chain triglyceride supplementation, which is a type of which is a type of fat. So they want us to see its impact on cognitive function before and after exercising. So to do this, they took a 2-week study where individuals either consumed the MCT oil or placebo every day, and then they would exercise for an hour directly after. So there were several cognitive tests that were given before and after exercising, such as memory and processing speed. So it was found that the individuals who took the MCT had better test scores than the placebo individuals and saw less mental fatigue. And the study concluded that the MCT was able to reduce fatigue in exercising and help the brain have a better performance for a longer period of time. So my methodology was that I first conducted a literature review, then followed by looking at the data and studies presented in the articles. I used a secondary source literature review to analyze information. I conducted both quantitative and qualitative analysis on data and findings regarding the effects of macronutrients and neuroendurance. And then I organized my paper into 3 sections: carbohydrates, proteins, and fats, and then I summarized my findings within each section. So I had several limitations while conducting my research. Some of these studies only focus on energy drinks and liquids with these macronutrients, not necessarily food. So the studies focus on carb beverages and not foods with carbs. We normally consume carbs in foods, but the research mostly focus on carb drinks. So this might not be a full representation of what athletes eat in a day. And then there were also some studies that showed contradicting points, such as that some of these macronutrients did not really have a big impact on neuroendurance. And then my last limitation was that it made it difficult to see if protein itself had a direct impact on neuroendurance, as the studies I looked at had the combination of proteins and carbohydrates. So, in conclusion, macronutrients do have a profound impact on neuroendurance, and they can directly improve cognitive function and reaction times. Carbohydrates were found to improve cognitive function and motor skills. Protein was able to improve muscle repair, and fats were able to decrease fatigue in exercising and improve the brain's performance for a longer period of time. So I just want to give a big thank you to Professor Varkey for giving me a solid foundation and background on neuroscience, as well as Coach Jen for her continued support of my writing process. And then I want to thank Coach Joe and the whole Gifted Gabber team for the opportunity to do this program, as I've learned so much through it. So thank you.
Excellent, Ayan. You want to stop sharing so that I can get Professor Vark?
Very
Yeah,
good,
go ahead.
clear presentation. Very good, clear presentation, I am. And I find that you are just in my neighborhood, but not only in my neighborhood, but the same school where my son goes to. So
Oh,
he's also a
really?
freshman at Yorba Linda High School. So I don't know whether you know him, Nathan Worthy. He plays for the soccer team. So
Okay.
anyway, um, so it was a good presentation. Question for you: do you think, like, how do you think, uh, like having these macronutrients? When I say macronutrients, just like having good eating habits, let's say, uh, would help with the cognitive function? Or, I mean, we know about the general body, but like about the cognitive function, why it's important? Why do you think, like your own thoughts?
Yeah, I mean,
After
obviously,
the,
yeah.
like the neuroscience lectures that you listened to.
Yeah, obviously, even what I found in the lectures that you taught, certain things can obviously affect the brain. So I looked at obviously the carbohydrate intake and different things like that, but that was able, it obviously gave energy to the brain, and then it helped with sports directly, because obviously if it was able to affect the brain, it helps with the cognitive function and reaction time, which is obviously crucial in sports. So yeah, I feel like that was,
Yeah, so because with the growing brain, or like even for the continuous wear and tear,
Mhm.
that our body undergoes. We should know, like our plasma membrane is mostly like made out of lipid. So we need a lot of fat. We need carbohydrate for energy, the ATP, as it was mentioned, and then we need protein for repair, continuous repair what happens in our body.
Yeah,
Good job, Anna.
thank you.
Yeah, a question from Professor Jen.
So great job on your presentation, Ayan. So you have clearly worked hard, and your findings really bring important and new perspectives to the field. But I have one question. Since you mentioned that you employed qualitative and quantitative method. Now,
Mhm.
in the quantitative portion of your study, how did you measure endurance performance, and what specific metrics did you track to assess, the impact of macronutrients? Or maybe how did you use quantitative, or what tool did you use for your findings?
Yeah, so I just in the articles I looked at the quantitative findings. It gave me like the specific percentage of either the drinks or how much it affected the brain. And then I used the qualitative like I try to summarize it and make understanding of it. But then so to answer your question, basically looked at I wanted to see specific articles that focus on the macronutrients and their effect on neuroendurance. And then basically in those articles, it told me the correct percentages for how it impacted brain and cognitive function.
But yeah, based on your answer, so it's not actually quantitative. You are just doing a qualitative method. Though there are percentage that you got from those existing literature, from those articles, but you are just like synthesizing, analyzing the information that you have gathered. But it is pure qualitative and not quantitative, because it has to do with the application statistical tools. But no, it's okay. The important is that your paper is well structured and very impressive.
Thank you.
Congratulations.
Thank you.
Excellent. So is this also NCT oil that you kind of chug for the keto diet? Is that the same thing?
I think so. I'm pretty sure that's what it is. They just said they took the MCT oil, so and
Yeah.
then yeah, so I'm pretty sure that's what it was.
Okay, so I'm gonna try that, and I'm gonna blame you if it doesn't work. I do have
Okay.
MCT oil. I'm gonna try everything that I learned today. Okay, so great job, Ayan. Uh, I mean, I don't have to go anywhere. I just have to come to the thesis defense, and boom, I learn a lot of things. So a wonderful job, everyone. Each and every one of you have contributed tremendously, and you've done amazing. And please don't stop here. Like I mentioned, you've got to take your thesis defense to your counselor and your teachers to get those counselor recommendation and teacher recommendation. Absolutely, because a counselor does not remember your name. Remember, they have 250 students. So I want you to take some time, go and tell them that, hey, I did research. I just need 4 minutes to share with you. Please do not share your research paper. Nobody has time to read that. But the same presentation, go and give it to them. They're going to remember, and it's going to reflect in your counselor recommendations. Got it? Everybody's got to do that. Okay, now let's go to Sohan Vendra. You're here, Sohan?
Yeah, I'm here.
You want to turn on your video, honey?
Sure.
Okay, there you go. Let's start.
Give me 1 second.
Mhm.
Can you see my screen?
Yes. You want to do present and then present? Yeah.
Sure.
Okay, go ahead.
Okay, hi. My name is Sohan Vendra. I attend West Windsor Plainsboro High School North, which is located in New Jersey. This is my thesis defense. To start off, the title of my research paper is called "The Variation of the Amygdala's Activation to Different Levels of Stress." In other words, so this research paper is primarily based off of the response to stress levels, which include chronic, acute, and traumatic stress. So the objective here is to investigate how the amygdala's neural activation varies in low, moderate, or high stresses, which is typically measured in the perceived stress scale, also known as the PSS, and analyze its implications for mental health. Most children might say that they don't know what the PSS is, but I can guarantee they do because usually in like starting from the age of 12 or say, in the pediatrics office, usually doctors isolate you in a room and they give you this sheet of paper for you to fill out. It's typically 10 questions long, and you have to fill it out genuinely, which usually measures your stress levels and can be used in reports. So, usually, the questions that I use to like put into databases were how does the amygdala respond to stress? How can the PSS be used to determine stress levels? And other key factors such as gender, sex, age, things like that to see my results. And more about the PSS. So, most children might recognize it now because this is what can be used to determine one's stress levels. So basically, current research that is on databases and other literature reviews today is things like recent fMR studies confirm that chronic stress amplifies the amygdala activation. And based off of my research through many secondary literature reviews, I found that there's in the right amygdala, there's a primary activation there that usually has a significant growth or a significant change in size. That's shown in the that's included in the structural changes. But here's the thing, with this type of research, there's many weaknesses to it or even limitations, and this is due to the fact that in today's society, there isn't like a foundation or a specific way to measure stress. It's all based off of polls, things like these, like the PSS, the perceived stress scale, and it's not really direct. There's nothing you can directly do to the brain that can actually measure your stress. Same with other neurodegenerative diseases or even PTSD, which is more relevant to stress in general. Other limitations can be like and to reiterate, like demographic gaps. This would include age, gender, differences in stress amygdala relationships. So in my methodology and results, I use this database called the FSL, which is the fMRIB software library to determine key changes in amygdala activation between types of stress, chronic, traumatic, and yeah, normal stress. So PSS scores basically how it's taken from in doctors in the pediatrics. So they measure it through moderate, high stress, or normal, where it's just moderate, high, and like normal. So moderate's like 0 to 13, then 14 to 26, and 27 to 40. And based off of that, they can diagnose you with depression or any other mental illnesses that, yeah. And for my statistical tools, since this was more of a quantitative research, I used Pearson's equation to correlate my PSS scores to the amygdala's activation. So my data that I used for the PSS scores was taken in 2021, and it took place. So obviously during this time, a pandemic took place, COVID-19, and it was taken in Saudi Arabia, fortunately not America. So then I found out that yeah, there's a right amygdala dominance, meaning that there's chronic stress linked to the larger right amygdala volume showing its growth. And specifically pertaining to the data, 30.2% of students had experienced high stress range showing like heightened amygdala reactivity, with 55% with moderate stress had intermediate activation. So now coming to my conclusion, here's the thing with taking the with children or even any adults taking the PSS, they could fill it out with bias and they might not be genuine. Even though you're isolated in your room, people might children might be afraid of filling it out truthfully, knowing that their parents might say, "Why is my um child depressed or even stressed? What have I been doing wrong?" So they might not fill it out truthfully always. And secondly, it's a limited sample, meaning it might not pretend like the data or the conclusions that are taken might not pertain to all ages, genders, and so forth. So contributions that were made throughout this research process was that we established the fact that like chronic stress may cause like many measurable physical changes in the amygdala, typically the right amygdala, which supports its role in stress-related disorders like PTSD. Okay, I want to thank Professor Varkey for the amount of time he put into the lectures and making sure everyone got the idea down for like many neurodegenerative diseases, as well as I want to thank Coach Jen and answering any question I had, especially on the one-on-one sessions, and organizing like all the ideas that you should have on your research paper. And lastly, I wanted to thank like Gifted Gabber into making this research process like possible. Thank you.
Okay, you want to stop sharing so that I can get Professor Varkey?
Sohan, very good presentation. It was very clear, and you did quite a bit of analysis as well. I think, like, that is very impressive. So now we know that, okay, amygdala is involved in stress. Do you do you think like you can also like think about other areas of the brain which is involved in stress? And do you think that you're going to work on the other regions of the brain as well, like the way that you worked on amygdala?
So yeah, from what I found, like the amygdala, the organ, like it's responsible with like specifically emotions. Maybe next time, like I wanna probably look at the hippocampus because I know like Alzheimer's negatively affects its like memories and things like that in the brain. Not only starting like from an old age, but even like the middle, maybe say like from 50. So I want to see how stress could also affect the hippocampus and maybe other organs in the brain.
Excellent. Very good job.
Great job. A question from Professor Jen.
So excellent job on today's presentation, Sohan. Your research is well thought out and thorough, and you presented in a very organized and approachable way. You explained everything and very specific. So my question is that very general, like how do you think your study could influence current practices or policies in the field?
That's a great question. So, what I was thinking, so by making conclusions about what changes can be done to the amygdala, I was trying to get at like how many people in like society today can like stop doing things that could negatively like harm what they're doing, can like negatively harm like their brain. So that could like reduce the chances of getting neurodegenerative diseases. Like, firstly, it obviously starts off with stress, and then it may go to neurodegenerative diseases. So by like reducing these chances could definitely help in the long run.
Wow, impressive. Congratulations again.
Thank you.
Okay, great job, everyone. I mean, every single presentation is amazing, and I want to give a big round of applause for all the hard work that went in. I know you must be like, my parents got me into this. I didn't sign up, but still, you guys did fantastic. Great job. So let's go with, hold on a minute. Nikita? Nikita?
Hi, give me a second to share my screen.
Sure. Yeah, a question for Sohan, please, and everyone else. Very, very interesting topics, and we want you to continue with this, not just stop. This is just the beginning. Okay, let's get started, Nikita.
For
Sure.
some reason, it's like not letting me share my screen. It's like there's like an error or something. It says like,
Okay, you want to put the presentation on the chat? I'll share, and you can present.
Okay.
Is it on Canva, PowerPoint? Which one is it? Okay, Nikita, I'm going to go to somebody else. You can just share the presentation on the chat and I'll be able to like present it. Okay, so let's go. We have quite a few. Aditi, you're here? Aditi? Aditi? Or let's go with Aishwarya. Aishwarya, you here? You wanna? Okay.
Hi,
Yes,
can
I've
you hear me?
started you. Yes, I can see you. Go ahead.
Okay, perfect. Let me just share my screen.
You're looking pretty.
Can you see my screen?
Yes.
Okay. Perfect. So my name is Aishwarya Ashok. I'm a junior at Central Bucks High School South in Pennsylvania. So just a little bit about me and why I came to choose this topic of modeling astrocyte influence on risperidone response in neuropsychiatric treatment. So I've always been interested in neuroscience in general. And a couple of years back, my dad and I read this book called The Root of Thought by Andrew Koob. And he's a professor at UConn, I believe. And he talked about how a specific type of cell in the brain called glial cells have a big impact on behavior and may have to do with the physical seat of the mind. And I was really inspired by that, and that's how I came across neuroscience in general. Now, my interest in this topic goes beyond just academics. As someone who plans on going into the military and pursuing psychiatry and treating people with disorders like post-traumatic stress disorder and even schizophrenia and bipolar disorder, I feel that it's really important for me to learn more about what I'm putting myself in the future to, and I really enjoy this topic. So let's go right into it. So first of all, I just want to give a brief introduction on what risperidone is. So risperidone is an antipsychotic drug used in schizophrenia and bipolar disorder. Now, bipolar disorder affects 5.7 million Americans, adults, just every year. And schizophrenia also affects 1.1% of the population each year. So these disorders and neuropsychiatric conditions have big impacts in our country and worldwide. And it's important to understand how to better personalize treatment for people who are suffering from these illnesses. So how does risperidone work? It works by competing with dopamine, the feel-good hormone of the brain, and works against it. So instead of dopamine binding to the dopamine D2 receptor, risperidone binds instead, suppressing overall dopamine levels and soothing the effects of schizophrenia and bipolar disorder. Now, there is a lot of variability with this drug despite its common use. A lot of patients experience weight gain, drowsiness, psychomotor agitation. Some patients reap a lot of benefits from using this drug, while others don't even see any effects from it. People also are not able to use this drug in conjunction with a lot of other drugs like high blood pressure medications, things like that. Sometimes you're not allowed to use risperidone with that. So there's a lot of variability, and our goal right now is to understand how we can better improve risperidone's efficacy and pull back from its side effects. And that brings me to astrocytes. Now, astrocytes we think of they're just another type of glial cell known as support cells in the brain. We kind of like to think of them as the janitors of the brain, clearing up extra neurotransmitters and other like calcium ions, things like that. And they may play a bigger role in neuroplasticity and supporting the neuron than we do know. And they also, research suggests that they may play a bigger role in our behavior. And that brings me to glutamate. That's our biggest focus for today. Now, glutamate is the primary excitatory neurotransmitters. So what that means is that it's basically like the gas pedal of the brain. So when you push on that gas pedal and release glutamate into the synapses, the neurons get really excited and they get stimulated, and that promotes activity. However, too much acceleration, too much of pushing on that gas pedal can lead to too much glutamate, and that's what we call excitotoxicity, and that can lead to neuronal damage and even cell death. So this is basically, I just wanted to show a little bit of a depiction and an example of how we can think of what's going on in our brain right now. So think of astrocytes as the traffic controllers, and risperidone is a bus trying to park into the D2 dopamine receptor parking spot. Glutamate is this traffic that astrocytes control. Now, when glutamate is properly controlled by astrocytes, we're thinking that risperidone may have a better ability to bind to the dopamine D2 receptor and fit into that parking spot. However, if there's too much glutamate going on, too much traffic, or astrocytes are not able to properly control the traffic, there may be an issue with the efficacy of risperidone binding into that D2 receptor. So that led me to the question I asked: how do astrocytic mechanisms influence risperidone's efficacy in neuropsychiatric treatment? And I hypothesized that variations in astrocytic glutamate uptake could contribute to the variability in risperidone efficacy. So I split my research into 3 phases: information gathering, computational model development, and simulation. So, phase 1, information gathering. I conducted a literature review, trying to understand what mechanisms were involved in risperidone, what mechanisms were involved in astrocytic functions, and what neurotransmitters I wanted to focus on. And then I identified what main pathways I wanted to look at, the main reactions that I wanted to look at going into this. Phase 2 was my computational model development. So I chose computational model development. I chose to use a computational model because I didn't have access to a lab or, any of these fancy trinkets that we can, do anything in person. So I decided to use a open source software called Copacity, and it's a biochemical pathway simulation software. So it basically allowed me to test my hypothesis without taking any biological risks, or it was easily accessible, basically. So part of phase 2 was determining my reactions. So these were the reactions that I used. First one was dopamine binding to the D2 receptor. The next one was risperidone binding to the D2 receptor, and then just glutamate transport. So glutamate from outside of the astrocyte coming into the astrocyte. And all of these were modeled into my computational model. And then I was able to understand how these factors played into each other. So another part of phase 2 was determining what initial concentrations I wanted to use, and I used external literature data for that. I also had to determine the kinetic values, also using literature data. Phase 3 was my simulation. So I decided to compare 2 types of astrocytes: a normal astrocyte with stable glutamate uptake, sorry?
Could you hear me? Can you hear
Yes.
me?
Yes.
I think your presentation is stuck on the first slide.
Uh-oh.
I mean, I tried to, but somehow I was muted, so.
Are you able to see it? Here, I'll stop sharing and try again.
So it has to be in the presentation mode.
Can you see it again?
And now, can you put it in the is it in the presentation mode?
Yes.
On the right-hand side, lower, bottom, right-hand side presentation.
Yeah, I am on presentation mode. Let me try again. Can you see it now?
Yeah, now we can see.
Okay. All right, so I decided to compare 2 types of astrocytes, the normal astrocytes with stable glutamate uptake, and then dysfunctional astrocytes with absent glutamate uptake. And I conducted a 600-second time course analysis, and I was trying to track the changes of risperidone binding to dopamine over time with changing glutamate levels. So this is kind of like what the table of my simulation looked like. And these are my results. So as you can see, this is just how the concentration of glutamate outside of the cell over time. This was trial 1, which shows the decreasing glutamate over time, meaning astrocyte is doing its job properly. And the trial 2 on the right shows a constant level of glutamate, meaning that astrocytes aren't doing their job and there is no glutamate uptake occurring. And just a key detail about this concentration specifically, this concentration models excitotoxicity. This is the concentration that's seen to be excess in the synapse. So these are my results for the concentration of dopamine binding to the D2 receptor over time on both trials. You can see that there isn't visually a significant difference. And same with dopamine by the risperidone binding to the D2 receptor over time on both trials. There wasn't a significantly big difference with changing levels of glutamate. I also conducted a statistical analysis and found that there was no statistically significant difference as well. So this leads me to my conclusion that glutamate didn't directly impact the efficacy of risperidone. However, I realized over time through this research process that computational modeling can definitely provide us better insight into patient-specific variations. And I also learned that there may be more to the picture than I'm focusing on. There is a lot of variations in vivo neural circuits compared to what I was picturing on my computational model. So there could have been some inaccuracies. I also had challenges with finding a good computational model in general. I originally had used another model called BioNetGen, but it was too hard to understand, and I switched over to Copasi because it was more user-friendly and it was easier to work with. And I also had trouble looking for the right kinetic values, the right parameters to set. And I had to do a lot of digging through literature to make sure that there was something there. I had some even accurate data. And also, just again, there's a lot of complexity that we're looking at between astrocytes and neuronal interactions. It's really hard as people we've just opened up this Pandora's box of astrocytes and neurons, so it's hard to accurately depict that on a computational model. In the future, I plan on adding additional astrocytic pathways. Astrocytes are also involved in the blood-brain barrier and things like that. And I want to investigate more on how we can personalize treatment with computational models. And finally, there's other neurotransmitters that risperidone is involved with, including serotonin. So I would like to add that to add more data and more complexity to my computational model. So these are my references. And I just would like to thank the Gifted Grabber program on giving me this opportunity to do a literature review and a quantitative analysis on a topic that I'm really interested in. Thank you for Professor Varkey for giving us your time over the weekends and helping us out understanding neurodegenerative diseases a little bit more. And thank you for Coach Jen for helping me out with my actual research paper and helping me out not only in this academic setting, but also giving us some information on how to improve in the real world with organizational skills. I would also like to congratulate everybody else who worked on their own projects along with me in this batch. So thank you very much. Any questions?
Yep. Excellent. You want to stop sharing? I'll have, yeah, and sorry about your presentation being stuck. I couldn't even make out. I'm blind. Sorry about that. Okay. So Professor Jobin, a question for Aishwarya.
Aishwarya, good job. It was a very good presentation, and I also see that you have put quite a bit of effort in understanding something new,
Yep.
for for high school level, that's really impressive. And also, like, well, just one question for you. So, since you did not see any difference, uh, could you think of why your experiment would not have shown the difference in the computational model, and why it still could be relevant in an actual wet lab experiment?
Of course. So Copassi is very versatile. It does give a lot of people opportunities to do research in a lot of different aspects, not just neuroscience. And so I believe that it wasn't necessarily an accurate model of in vivo neural circuits. Um, our brains are so complex, and there's so many interactions happening all at once, and it may have not been the most detailed or focused on um, astrocytic and neuron interactions. And we talked about this a bit earlier. It I did a 600-second time course analysis, so it was 10 minutes. But in reality, what goes on can happen in nanoseconds. So I could even zoom in a little more and see if there were any drastic changes in a more smaller time period.
Very good answer, Ashit. Good job.
Thank you.
Excellent. A question from Professor Tuban.
Yeah, congratulations on delivering such a well-rounded presentation, Aishwarya. And it's clear that you have invested a lot of time, energy into your research, and the way you present it to us, the results speak for themselves. Anyway, this kind of question is like this is similar to uh, to Professor Varkey's question. Like, how do you see computational models helping to expand our understanding of how risperidone interacts with other neurotransmitter system, and then what specific interactions would you like to investigate next?
Of course. So, just to answer your first question, um, computational models are very important in helping us do research in without having to use actual people. And it's really hard. And some people talked about this earlier, but neuroscience research can be difficult without using invasive strategies to really understand what's going on. And so, a computational model can really help us understand chemical reactions and the kinetic pathways that are going on, and zoom in without having to use participants. And um, in the future, I really want to use serotonin as well. And I want to look at serotonin because risperidone also works to block serotonin levels. Um, that also cause symptoms of bipolar disorder and schizophrenia. And so these this schizophrenia this the serotonin could also be another aspect of how risperidone can influence holistically and not just looking at glutamate. Glutamate is also impacted with calcium signaling. And so I also wanted to look at that, maybe seeing how calcium signaling and glutamate and astrocyte glutamate uptake all work together to help risperidone be more efficient.
Wow,
But yeah.
impressive. Congratulations.
Yeah, thank you, Aishwarya. Aishwarya also does Thrive, where she mentors kids to overcome procrastination. So amazing job, very good. We could have split the number of people. I'm sorry about the wait, but it's very, very interesting. Let's go with Arnav. Arnav, are you here?
Yes,
Arnav?
hi, I'm here.
Okay, you want to go?
Yeah, sure, no problem.
Okay.
Um,
Yeah. You
hi,
want to put it in the presentation mode so that, yeah, great.
hi. Hi, I'm Arnav, and I'm an 11th grader. I attend Liberty High School in the state of Washington. And today I'll be presenting my topic on the gut-brain axis and how it can serve as a new frontier in the treatment of depression. So just to introduce, I'm investigating on how gut microbiota impacts psychological, physiological health, and particularly depression. My research question focuses on how the gut dysbiosis, which is the imbalance of microbiomes in the gut, and how they influence mood regulation and depression through the gut-brain axis. This is really important because it's important to understand this because it can help develop new treatments for patients, the minority of patients who are unresponsive to current therapies. So my objective is to explore the link between microbiota disturbances and mental health disorders. I also just want to point out in the diagram here that uh, the imbalance of microbiomes leads to the dysregulation of neurotransmitters, which can lead to stress and increased anxiety. So in terms of the literature review, I gained a depth of knowledge from this, but there are four main things that I just want to point out. So I learned that gut microbes produce key neurotransmitters like serotonin and GABA that influence mood and cognition. So serotonin is famously known as a major mood enhancer, and GABA is the most inhibitory neuron. And also dysbiosis disrupts the gut lining and increases pro-inflammatory cytokines such as interleukin-6 and TNF alpha, leading to depression. Additionally, studies have also showed that reduced levels of lactobacillus and bifidobacterium in individuals with depression. So lactobacillus and bifidobacterium are actually considered good bacteria, so the reduced levels leads to increased depression. And finally, inflammation and microbial imbalance are linked to HPA axis disruption and emotional dysregulation, which increases depression. So in terms of the methodology, I took it from a quantitative standpoint and a qualitative standpoint. So I reviewed over 15 peer-reviewed articles and meta-analyses from PubMed, Google Scholar, and JSTOR. Qualitatively, I made sure to organize my findings psychologically, physiologically, and metabolically so that I could cover a broader amount of information. So some key findings that I found was increased cortisol, which leads to increased stress, leaky gut, reduced shorty chain fatty acids, mood and cognitive disturbances, and metabolic changes like insulin resistance. So also some treatment options are that I found out is prebiotics, so digestive fibers in the starch and glucose. Probiotics, you can get it from yogurt, such as beneficial bacteria, and fecal microbiota transformation. And all of these stabilize the gut microbiome and help alleviate the symptoms of depression. So in conclusion, my research question, it just explores how the gut dysbiosis, the imbalance in the gut microbiome, affects depression by disrupting the gut-brain axis through inflammation, neurotransmitter imbalance, and just emotional dysregulation. Some of the key contributions. So I feel like this research highlights the gut microbiome's role in mental health and suggests that targeting it with treatments like probiotics, prebiotics, or dietary changes may offer promising alternatives, or it could be used adjunctively with current antidepressant medications. So, however, some of the limitations that I found through my research was so much of the current evidence is used on animal models or like early stage or phase human trials. So there's not much, there hasn't been much research shown in the long-term effects and standardized treatments. So it requires more research. And also this is a literature review, so there was no additional research that was being done. So before I conclude, I just want to thank Dr. Varkey, Dr. Jennifer, and Mrs. Joe and the entire team of Gifter Gabber. Dr. Varkey and I, we had sessions together and he kind of guided me through how to review peer articles, and it was really insightful for me to continue my research. Dr. Jennifer, she like guided me through the process of constructing the research paper, which made the process much smoother. And finally, Mrs. Joe and the entire Gifted Gabber team, because they provided me the opportunity to interact with these mentors and help me project my voice as a student researcher. Thank you.
Okay, you can stop sharing. I'll have Professor Varkey ask you a question. And Professor Varkey, would you like to adjust the volume a little bit?
high volume, the mic.
Yes.
So can you hear now better?
Yeah.
A bit louder.
Yeah, see my mic, I think like after this. Just
Okay.
to improve,
No problem.
like maybe the system volume is less. But Arnav, like thank you very much for your presentation. It was very good and focused and also like very timely. Just wanted to let everyone know that, yeah, this is like one of the hot topics right now, the gut and brain axis, how they're linked to each other. And so in your study, did you, Arnav, find any connection between like certain eating habits and depression or mood disorders?
Yeah, so certain eating habits, kind of how I mentioned with the probiotics. So
Uh-huh.
these like eating yogurt, it can help prevent depression because it serves as a probiotic, and these are kind of like the treatments that are being explored right now. And these treatments are helping to like eventually like prevent the imbalance of microbiota, and preventing that imbalance of microbiomes can also just lead to like more like regulated emotional regulation. So it maintains the body system. So it definitely like limits any like spike in depression. So yeah, there was research on that, yeah, a little bit.
Very good job. Arnav.
Yeah. A question from Professor Tuban.
Yeah, you know what? That was an excellent and detailed presentation, Arnav. The clarity which, you presented complex information is highly impressive. And yeah, your paper is very clear and well organized, and there's really nothing more to ask about the content. But I have one question which is general. What was the most challenging part of the research process, and how did you overcome it?
Yeah, so definitely the most challenge. So there were kind of like a couple of challenges. Also, so this my study kind of involved like immunology and neuroscience. So like this is like interdisciplinary. So it's kind of like from the articles that I was trying to gather and the review articles. I had to find articles that like specifically kind of suggested any connection, because these fields are like in the field of science, they're interconnected, but they're also like really different. So it was definitely hard to initially when I was trying to find those review articles, it was definitely hard to like find the connection. But eventually I was, and yeah, that was one of the challenges. Yeah.
That's fantastic. Thank you, Arnav.
Yeah, wonderful. Thank you for sharing your presentation. I'm learning quite a bit I mean, pretty much every discussion here is about anxiety and depression and brain health. I feel like I have to apply everything. But thank you so much for coming here and presenting. A great job, everybody. It's almost as if I'm in a room of PhD candidates, and I have to remind myself that you guys are high school students. So it's wonderful. I want to thank Professor Varkey and Professor Tuban for working so hard. Yeah, so let's go ahead with, oh wait, we have Nikita. Okay, so yeah, you can, yeah, Nikita, you here? Okay, I'm just gonna, okay, 1 second. Okay, I'm gonna share my screen. You can present. Okay, go ahead.
Good afternoon, everyone. My name is Nikita Arun, and I'm a sophomore at Coxsville High School in North Carolina. I just want to thank the Gifted Gabbard team first for providing me with such a wonderful experience learning and researching on neuroscience for the past 3 months. I have learned and gained so many valuable skills with like creative writing and much more. The research topic I chose today is the role of opioids in treating Alzheimer's disease. Um, the research that I conducted focuses on the relationship between opioid use and Alzheimer's disease and the complexity involved in it. While predominantly associated with increased risk, opioids may affect It may offer benefits in managing certain Alzheimer's disease related symptoms. This presentation will explore those nuanced areas. So Alzheimer's patients can experience pain from camoroid conditions. Opioids can provide. Can you go back to slide 2? Sorry. So Alzheimer's patients can experience pain from sorry. Opioids can provide effective analgesia, improving quality of life. Pain relief may indirectly reduce agitation and behavioral disturbances. In some cases, opioids may have a calming effect, and this may mitigate severe agitation or aggression and unresponsive to other treatments. Opioid is not a first-line approach. Careful consideration of risk versus benefits is essential. In advanced Alzheimer's stages, comfort is the primary goal. Opioids can play a crucial role in managing pain and distress. Opioids can provide compassion for end-of-life care, and opioid use in Alzheimer's patients must be approached with extreme caution. Significant risk can include cognitive impairment, increased mortality, and adverse effects. Benefits must be carefully weighed against risk. Opioid therapy should be reserved for specific, carefully monitored situations. Can you go to the next slide? So the opioid system may contribute to abnormal of Pau protein, which leads to neurofibrillatory tangles, another key pathological factor of Alzheimer's. The opioid system has a complex relationship with amyloid beta or alpha beta production. Dysfunction of opioid receptors can increase alpha beta production by hindering breakdown with endocytosis and degeneration of enzymes, and gamma secretase, which is crucial for alpha beta formation. Conversely, activating opioid receptors can decrease alpha beta production by promoting the breakdown of these enzymes. The opioid system dysfunction is implicated in neuroinflammatory, a chronic inflammatory state in the brains associated with Alzheimer's. Opioid receptors can influence the levels and activity of several neurotransmitters that are known to be dysregulated in the Alzheimer's and contribute to its pathology. Opioid system interacts with alpha beta production through its effects on BACE1 and gamma secretase. This is a crucial point as these enzymes are directly involved in the formation of amyloid plaques, a defining characteristic of Alzheimer's. Its involvement is in multiple aspects of Alzheimer's pathology. The opioid system presents a promising target for the development of the new Alzheimer's treatments. Modulating opioid receptor activity could potentially offer a way to slow down or even prevent the progression of this disease. Can you go to the next slide? The interaction between MOR and DOR opioid receptors in the brain. Opioid receptors, these are G protein coupled receptors that play a crucial role in modulating various brain functions, including automatic, involuntary bodily functions, sensory perception, emotions, and cognitive processes. MOR and DOR interactions. The study highlights the MOR and DOR functionally interact within living organisms. However, the specific level which interaction occurs was previously unknown. The dataset was created by red MOR and /green MOR double knock-in mice. The genetically modified mice so that their MOR receptors were tagged with the red fluorescent protein and their DOR receptors were tagged with the green fluorescent protein. This allows the visualization of both receptors simultaneously. Further performed dual receptor mapping throughout the nervous system, meaning map the location and distribution of MOR and DOR receptors. This mapping was done with a subcellular resolution, indicating a very detailed level of visualization within cells. The data was organized into an interactive database and made accessible online as a opioid receptor atlas with the ability to visualize MOR and DOR together and also use co-immune precipitation, a technique used to identify protein-protein interactions to provide evidence for receptor heteromerization in these mice. Can you go to the next slide? You can see here the red fluorescent and the green fluorescent in the mice's brains. Targeting signaling mechanisms unique to MOR/DOR interactions could lead to the development of innovative therapies. Pinpointing brain regions where MOR and DOR interact at the intercellular level provides valuable clues for developing therapies with specific effects. While MOR is a primary target for painkillers, the development of opioid tolerance is a significant clinical problem. There's evidence suggesting that DORs contribute to morphine tolerance, but the precise molecular mechanisms of this MOR/DOR interactions are very unclear. Studies in laboratory systems have shown that MOR/DOR heteromers have distinct trafficking and signaling properties. Signaling these heteromers are considered a way to reduce opioid tolerance. The high co-localization, the presence of the same neurons of MOR and DOR in pain pathways supports the idea that heteromers are a good target for pain management. Neurons expressing both MOR and DOR was also found in the brain system areas, which connected to autonomic nervous systems. In these locations, two receptors may work together to cause physical and automatic symptoms of drug withdrawal. Co-expression of MOR and DOR in the lateral hypothalamus, particularly in oxytocin-producing neurons, has a interesting implication. MOR/DOR heteromers in these neurons may be a target for new treatments for obesity since oxytocin neurons are also crucial for drug reward, targeting MOR/DOR specific mechanisms in this area that could potentially reduce drug-seeking behavior. The next slide. While DOR activation promotes alpha-beta production, some studies have found increased levels of DOR in Alzheimer brains. This seemingly contradictory finding may suggest a complex content-dependent role for DOR in Alzheimer's disease. The brain might be attempting to compensate for other issues by increasing DOR, but the increased DOR then accelerates alpha-beta production. Despite the promising preclinical results, DOR antagonists have not yet been tested in human Alzheimer's trials. The text highlights the need to further research to explore the therapeutic potential of DOR antagonism in humans, while the text mentions earlier unsuccessful trials with non-selective opioid antagonists emphasizes the importance of focusing on opioid-specific reciprocator subtypes, particularly DOR, the different effects of MOR. Antagonists in different studies highlight the complexity of the opioid system. In essence, DOR is promising therapeutic target in Alzheimer's. The evidence suggests that blocking DOR activity could be a way to reduce alpha-beta production and slow down the progression of the disease. However, MOR opioid reciprocator in PS1 mouse model of Alzheimer's disease, we demonstrate that DOR activation by UFP512, but not MOR activation by DAMGO significantly impacted cognitive impairment, reduced beta amyloid production and aggregation, and protected neurons from APP mice. Furthermore, DOR and MOR exhilarated differential modulation of microglia in both PS1 mice and cell models. Specifically, DOR activation inhibited excessive microglial activation in 5-release pro-inflammatory cytosis, hallmarks of Alzheimer's pathology. Gene expression profiling revealed that DOR/MOR alterations were closely associated with microglial homeostatic signatures and high mobility protein B1 pathway in Alzheimer's. Next slide. In a microglial neuron control system, DOR overexpression in the microglia protected neurons from the apoptosis and reduced inflammation, likely limiting the HMGB1 release. The largest changes in MGB1 due to UFP512 treatment occurred in the microglia. The findings align with postmodern studies showing diseased MOR and DOR densities in the hippocampus and the cortex of Alzheimer's patients. This suggests potential therapeutic benefits of DOR targeted interventions for Alzheimer's. Previous studies using DOR antagonists showed improvement in Alzheimer's models, but this work shows the improvement of using DOR agonists. The difference is likely due to the age of mice used in testing. Older mice used in the study showed improvement with the agonist. DOR activation offers more robust and sustained neuroprotective effect against Alzheimer's than MOR activation. DOR beneficial effects are likely mediated through its modulation of microglial function and HMGB1 signaling. The relationship between DOR and C1q and microglia warrants further investigation. Further research using PLX5122, a microglia elimination drug, will be used to further understand the role of microglia in DOR mediated neuroprotection. The next slide. For my conclusion, the recent investigations have highlighted therapeutic potential of the delta opioid receptor in mitigating neurological stress. Substantial evidence supports the neuroprotective role of DOR activation. These findings position the opioid system, specifically DOR and its ligands, as promising avenue for developing novel neuroprotective therapies. To translate these preclinical findings into clinical applications, further translational speech is warranted, specifically in-depth investigations for DOR mechanisms of action are crucial for optimizing opioid-induced neuroprotection. Furthermore, exploring strategies in upregulate DOR expressions could enhance therapeutic efficacy. This research direction holds significant promise for addressing a range of neurological disorders characterized by neuronal damage and death. Next slide. I would once again like to talk about my research experience with gifted gabber. I just want to thank Professor Varkey and Miss Tubin. Sorry, my throat is really dry. I learned so much from this experience, and yeah, thank you. Next slide.
Sorry about that. Let me get Professor Varkey to ask you a question. Yes, Professor Varkey.
Can you hear me, Nikita?
Yes.
Is it better?
Yeah. Yeah.
Okay. So Nikita, that was quite a bit of effort that you have put in, and quite a bit of information there as well. I think you might have spent like several hours preparing this presentation itself. So just wanted to uh, say, like, can you, let's say, based upon all the receptors that you talked about and EBIT and everything, can you summarize your work in, let's say, a couple of statements? What do you think from your point of view, what you learned from this, and what can be done for future?
Do you mind repeating that? Your voice was like a little bit
Not
scratchy on.
clear?
Like it's like kind of clear, but it's like really quiet, kind of.
Oh, I don't know why it's happening. Can you hear now? Is it better?
Yeah, it's like kind of better.
Okay. So what I wanted to say is that can you summarize your uh, presentation in, let's say, a couple of sentences?
So limiting the participants are eligible for use with overuse or abuse of opioids, and in some cases, like opioids have like a calming effect, and my presentation is just basically about opioid use for Alzheimer's patients, and yeah.
Okay, so is it do you think that it can be used as a treatment in the future?
Yes, I do.
Okay, okay, good job.
Let's have Professor Jen. And I also want to let participants know that please don't read off your presentation. It's just summarizing it, couple of points, okay, to keep it crisp and like terse and effective. Okay, so let's go with Professor Jen.
Yeah, hi Nikita. Your presentation was impressive, though I noticed that the specific objectives of paper and the reasons behind choosing this study were not mentioned earlier, and could it be also found in your research paper and your research draft? So I would like to ask, what were the main objectives of your research? What did you aim to explore or to investigate, and why did you choose this study?
My the use of like I try to find like the positive use of opioids and limiting participants.
Okay, so maybe we can just like improve that. Okay, find, to identify, to determine. We can actually use powerful words, but yeah, maybe we'll just give you more suggestion, and we'll help you just maybe book a meeting with us to help you guide and to help you structure your paper well. Okay, congratulations still. Good job.
Wonderful. Thank you, Nikita, for that detailed presentation. Let's go to Prisha. Prisha, are you here? Yeah.
Yeah, I'm here.
You want to turn on your video?
Yep.
Okay. Okay, there you go. Looking all sharp. Go ahead.
Um, Sorry, it's not letting me share it on my own. Would you be able
Okay.
to?
I will. I'm just going to let someone go, and then you share it. Sometimes what happens is you need to set up sharing aloud. So just message me your presentation. Let's go with Sanvi. Sanvi, are you here? Yeah.
Yes, I'm here.
Yeah. You want to go ahead?
Yes. Oh, sure. Okay. You all see my presentation?
Yes. Excellent. Go ahead, please.
Okay. Um, okay, so hi, I'm Sanvi Valetti, and I go to Jordan High School in Fulshire, Texas. And today I'll be talking about the effects of music therapy on neurodegenerative and psychiatric disorders. So you may be asking, like, what is music therapy and how does it work? Well, music therapy is a powerful clinical practice that uses music to support the health and well-being of individuals. Music therapy isn't just about listening to music, it's like a guided therapeutic process that is led by trained professionals to help manage emotions and improve mental health and overall enhance the quality of their lives. So there are various types of music therapy that each offer unique ways to engage with music. For example, receptive music therapy, it involves listening to music and responding to it, either silently, verbally, or emotionally, and it's really good for relaxation and emotional release. The next one would be recreational music therapy. It's like a more interactive way in which clients are able to sing and play along with pre-composed music, and it's enjoyable and helps build confidence and communication skills. The next one would be improvisational music therapy, as it allows for spontaneous music making using instruments, the body, and the voice. It encourages emotional expression and creativity, with therapists adjusting the session based off of the client's mood. The next one would be compositional music therapy, as it focuses on songwriting, and clients create like original songs and lyrics with the help of the therapist, and it helps them express thoughts in a creative and meaningful way. So music therapy, it can happen one-on-one or in groups, and some approaches even include like guided imagery and support deeper relaxation and insight. So overall, music therapy offers like a various range of for people to connect with music and use it as a tool for healing and growth. So for my research on music therapy, I focused on three main sources that provided strong evidence and insight into how music can be used as a therapeutic tool in different clinical settings. So, for example, ONSUS and CAN in 2025, they explored the use of music therapy as a treatment for neurodegenerative diseases such as Alzheimer's and Parkinson's. Their study showed that music therapy can help improve cognitive functioning and emotional well-being in patients suffering from these conditions. This was especially useful in understanding how music therapy can support the brain and other memory-related functions. Another article that I use would be by Lassner and his peers in 2025. They explored the impact of music therapy on psychiatric disorders, specifically anxiety and depression. Their findings showed a significant decrease in symptoms, suggesting that music can be an effective, non-drug, and non-invasive based approach for mental health care. And this helped me understand how music therapy could benefit emotional regulation and psychological recovery. Last but not least, Ketcherpal in 2009, she focused on developmental disorders and showed that music therapy can improve communication skills in individuals with conditions such as autism and other intellectual disabilities. This source helped me broaden the scope of my research by showing that music therapy is not limited to just mental or neurological illnesses, but also supports developmental and social growth. So combined, these sources help shape the foundation of my project, providing like a well-rounded research and understanding of different ways music therapy is applied in real-life clinical settings. For my methodology, I used a meta-analysis approach, which means I reviewed and analyzed findings from multiple peer-reviewed sources rather than conducting original experiments myself. This allowed me to compare results across various research perspectives and papers to draw broader conclusions about the effects of music therapy on various conditions. And in order to get these sources, I used academic databases such as JSTOR and EBSCOhost, which are known for providing access to reliable and peer-reviewed journal entries. These platforms help me find up-to-date studies on how music therapy has been used to treat such disorders like depression and anxiety, as well as neurodegenerative and developmental conditions. By using these trusted sources, I was able to ensure that the information in my research was both accurate and evidence-based. So therefore, this method gave me like a strong understanding of the different ways music therapy is applied in clinical settings. In conclusion, Music therapy has been proven to be a non-invasive and cost-effective and beneficial form of treatment for individuals with psychiatric, neurodegenerative, and developmental disorders. It offers meaningful improvements in emotional well-being, in mood regulation, and even cognitive functioning. However, while these results so far are highly promising, it's important to note that there's still much more research to be done, and it will help refine the methods and standardize these practices and overall ensure that music therapy becomes an even more effective tool and therapeutic method. So a little bit about my research experience with gifted gabber. Working with Professor Tubin was extremely helpful. I received thoughtful feedback on my research paper, and it really helped me learn and take all the ideas in my head and organize them clearly into words. It was a great step in building my academic writing skills. With Professor Varkey, I had the chance to dive deeper into the world of neuroscience. I learned a lot about neurodegenerative diseases, which gave me a nice solid foundation and sparked my interest even more within the field. I also got a broader understanding of how neuroscience works in general. And overall, this research experience gave me a new perspective on the importance of research. It wasn't just about the information I learned, it was about seeing how the whole research process works from start to finish. And it really showed me how meaningful and valuable research can be. So, thank you.
Wonderful. You want to stop sharing so that Professor Abarki can ask you a question?
Yeah.
Tanvi, can you hear me? Just wanted to ensure that everyone is able to hear me clearly now.
I am able to hear you.
Okay. So, Tanvi, good presentation, very focused presentation. For you, like the question is like, could you tell me what happens in the brain? What are the pathways or what are the part of the brain in like particular regions which are affected when you listen to music?
So there's like various regions that can be affected. For example, the auditory cortex, the prefrontal cortex, the hippocampus, the amygdala, all of these, all these parts of the brain, they come um, to create like mood regulation and decision making and planning and even memory and like fear and pressure. It regulates all of that. So therefore, many of these disorders are linked to these issues in these parts of the brain. So listening to music can help alleviate these symptoms by regulating like disabilities in these parts of the brain.
Yeah, so basically, like turning on the circuits and then making those neurons active.
Exactly.
Good job, Sanvi.
Thank you.
Okay, great job. Let's have Professor Jen.
Hi, Sanvi. Your paper shows great potential, and I want to thank you for your excellent and a thorough presentation of your findings. It's very impressive, to see such a well-rounded paper. And I don't have major comments correction because it's so organized and clear. But I have this one question, like it's general though. If you were given more time and more resources, what additional areas would you like to explore to expand this study in the future?
In the future, I would like to talk about how it can affect not just neurodegenerative or psychiatric disorders, but how it can also play a role in other areas of treatment, for example, oncology or gynecology, as there has been instances in which um, music therapy has helped alleviate symptoms and caused a fighting mood for the patients.
Wow.
Okay,
Impressive.
great job. Let's go with Amia. Thank you, Sanvi. Amia.
I'm going to share my presentation. For some reason there's an error, but can I send you my link to the presentation?
Yeah, please. Anybody who can't present, please share your presentation. I will do that. Now let's go with Jordan. Meanwhile, you share the presentation. We'll quickly go with Jordan and then You can share your Canva. Okay, Jordan, you want to go ahead?
Yeah, sure. Can you see me?
Yes, I can.
Okay, let me try
Go ahead.
to see if my presentation works.
Okay, anyone else? Okay, let me just see if I can open this. Yes, I can open Amia's. Okay. So Amia, you can go next. Yeah. Jordan, can you click present and present?
So can everyone see my screen?
Yes, go ahead, please.
Okay. So my name is Jordan. I'm a sophomore. I live in Massachusetts and I attend Lexington High School. So my topic of my research paper was the impacts of mutations in the TP53 gene specifically, and how that impacts the cell cycle and cancer development. So for an introduction, obviously my topic areas are going to be mainly about cancer, the cell cycle, and the TP53 gene specifically. So one crucial question I wanted to answer was focused on the solutions of the negative consequences of cancer and how a mutation in the TP53 gene would actually affect the cell cycle. And I wanted to research how scientists are currently fighting against these mutations and what we can do in the future. You might ask yourself, why is this important? Why do we have to care about this? Because TP53 mutations are the main cause of 50% of cancers. So if we want to try and fight cancer as a whole, fighting again and discovering what's important in TP53 would be really critical in this field. So for my literature review here, I just provided 3 of the articles or research papers I used when I was trying to research and make my own paper. So the findings of each of these, I'll just go over them. For the first one, we can look at cell regulation in normal conditions when there's no mutated P53. And this was useful in understanding how, in building an understanding for myself as I could learn about how the cell actually regulates, how it uses cyclin and CDKs to actually have checkpoints and prevent mutations from proliferating, as we see with the cancer. And then another useful article was this second one by Katrian Vermeulen. It talked about the therapeutic approaches, which is what scientists are currently doing to actually fight against these mutations. And as in my paper, I talked about these therapeutic targeting and approaches and how these are the future of fighting cancer and fighting mutations in the TP53 gene. And finally, that last article talks about just the domains of the P53 protein and how it works and how, and using that understanding, I could learn about how mutations in that actual in each domain of the protein could further proliferate any sort of tumor growth or any issue. So for my methodology, I relied mainly on studies from sources like PubMed and JSTOR, and also clinical trials on what's going on for these therapeutic approaches. Because right now, these therapeutic approaches are just being discovered and investigated in. So looking at these clinical trials that are occurring through these articles on PubMed, I could learn about how scientists are fighting. And then obviously I use other articles which were useful in gaining an understanding about how this whole cell cycle works and how mutations actually happen. And finally, I also learned about what are gain of function mutations. These are pretty important because it's one of the key reasons why cancer becomes something that is so aggressive, as these mutations, which are unlikely to happen but can happen because we have just so many cells in our body, can lead to a gain of function through many of these cells having the ability to proliferate and lose their density dependence and be able to grow without any limits, which leads to tumor growth, which could be just a benign tumor, which doesn't lead to anything, or could be very cancerous. This relates to the third point I learned about how cancer actually develops. In my paper, I talk about how cancer like affects a person and how it will start in just a singular cell but could spread throughout many different processes through going through the bloodstream and going to a remote area of the body and spreading cancer in that area. And then the main significant finding was the treatments that are going on right now with these therapeutic approaches. There are various treatments that are happening with these clinical studies, as I said, and learning about them was very interesting. And I wrote about them towards the end of my paper. And so for my conclusion, I'll remind you my topic was mainly about these mutations and their impacts on the cell cycle and cancer development. And so in my paper, I tried to like differentiate the impacts and talked about first how the cell cycle is like regulatory effects are affected and how cancer will actually proliferate after like these mutations because of various reasons. And something like why my impact on writing this research paper actually matters is to further therapeutic approaches as a whole. When I was writing about and understanding all these therapeutic approaches, I tried to convey them in a method which allows other people to understand what is like these therapeutic approaches that are happening right now and further events or further findings that could happen. And for my limitations, obviously my paper was a secondary literature review, meaning that there are going to be some limitations. I wasn't conducting these actual experiments. And then also the fact that since it was a meta-analysis where I used like multiple pieces of evidence, multiple studies, it led to the studies not having the same criteria, which which could affect the results that I found. And so that's obviously one of the main limitations. And then some other limitations are other important factors that could occur and affect these results. These could include like the environment which these like studies happened in, or the environment of the people who were studied happened in. And like that's just one example. But when we're studying and understanding all of these factors, we have to sort of take into consideration that there's like other important factors that aren't in our control. And as a lasting note, I would just like to say thank you to everyone, a part of the Gifted Gabber community, Coach Joe, especially thank you to Dr. Varkey and him and the one-on-ones that we had when he helped me understand how I should like write my whole paper. And then especially a thanks to Professor Jen, who helped me at the start when I was trying to understand how my paper should be structured and how I should sort of like how I should just phrase it.
Yes. Fantastic. Great job, Jordan. You can stop sharing. I'll have Professor Warkey ask you a question. And also, please don't think that you're on the Titanic waiting for a rescue boat for your turn. I'm sorry that it took so long. We are all learning here. I want you to enjoy everybody's research and not look at the clock. I know it's hard. We are almost there. So, Professor Warkey, a question for Jordan, please.
Presentation, Jordan. You have done a very focused research, although it's not in neuroscience, but I'm glad that I could still help you. So a question, Jordan, for you. Do you think that any mutation or every mutation that happens in P53 can result in cancer?
No, like there's these different types of mutations, like missense mutations or like silent mutations, where like even if it's just a singular like nucleotide changing, it won't lead to any amino acid change, or even if it's a singular amino acid chain, it's not going to lead to a difference in the way that the protein folds, which won't affect the actual P53 protein. So as I said in my paper, I talked about like 3 specific amino acids that could, if they were to be changed, lead to drastic impacts. So to answer your question, I don't think that every single mutation or singular mutation could affect the whole like P53 and lead to cancer, but only a few specific minute ones could.
Very good answer. Good job.
Thank you.
Professor Chuban.
Yeah, thank you so much. You did an excellent job, Jordan, presenting your findings in a logical and very comprehensive manner. Also, your work shows great attention to detail, and it's clear that you have dedicated a lot of time to really to produce this paper. Now, my question is, what do you believe is the significance of your paper in advancing our understanding of TP53 mutations and cancer development? Also, um, what recommendations would you give to future researchers who are also interested in your study?
So to answer your first question, I think my paper plays a role in understanding how these mutations actually happen and how they don't not only affect in cancer but also the whole cell cycles regulation process. And I think the main way that it could act as like a recommendation is through like its therapeutic approaches. That sort of section in my research paper, which talks about like future like innovation that could happen, because in my paper I talk about like the current situation of these like innovations that are happening, but for any sort of future researcher they could try to learn from my paper and understand how it's going on right now, how it actually functions, and try to innovate based off that.
Very good. Thank you for that.
Excellent. Excellent job, Jordan. And you're an excellent presenter. Wonderful, very enjoyable. Two questions for Jordan. Let's go with Amia. Amia, I can open your presentation. So let's get started. I'm just going to share and you can tell me when to change. Okay.
Okay. Today, I'll be presenting my research on the impact of estrogen fluctuations on mental health. My name's Amia Sidhu, and I'm a sophomore at Portola High School in Irvine, California. Okay. A little introduction, specifically how changes in estrogen throughout a woman's lifespan can influence mood disorders such as depression and anxiety. The central research question I set out to address is how do fluctuations in estrogen levels throughout a woman's lifespan impact her mental health, particularly anxiety and depression. To investigate this, my objectives were to establish the connection between hormonal fluctuations and psychological health by reviewing current literature, and then also examine the relationship between estrogen changes and mental health conditions, particularly focusing on how these fluctuations could occur during key stages in a woman's life, such as puberty, pregnancy, postpartum, and menopause. And then 3, to identify possible mitigation strategies or like interventions. Okay, literature next, please. Okay, moving on to literature review, a significant amount of current research has been explored. The link between hormonal fluctuations and mental health also. Studies consistently show that estrogen plays a key role in regulating neurotransmitters like serotonin and dopamine, which is crucial for mood regulation. For example, several studies have shown a clear relationship between decreased estrogen levels during menopause and an increased incidence of depression. One key theory I examined in my research was neuroendocrine theory, which posits that fluctuations in hormones, especially estrogen, can lead to changes in the brain, thereby influencing mental health. This theory supports the idea that hormonal change during critical life stages contribute to mood disorders. While much of my literature focuses on the effects of estrogen during menopause, my research expands this to other life stages such as pregnancy and puberty, areas that are often unexplored. However, a major weakness is the current body of research because there isn't a lot of long-term studies to track hormonal changes in a woman's life, so it could limit the validity of my research. Okay, next. Now let's discuss the methodology and results of my research. To analyze the connection between estrogen and fluctuations in mental health, I employed a qualitative secondary review of literature examining peer-reviewed studies, clinical clinical trials and meta-analysis published in reputable journals. I analyzed data regarding how estrogen levels fluctuate during different life stages, such as puberty, pregnancy, postpartum, and menopause, and how the fluctuations correlate with reported incidences of mood disorders, such as depression and anxiety. Through this process, I was able to identify several recurring patterns and connections, providing a deeper understanding of psychological impacts of these hormonal shifts. The key results of my analysis show a significant correlation between lower estrogen levels and an increased risk of developing anxiety and depression, particularly during menopause and after childbirth, because low estrogen levels create low serotonin and dopamine levels, which makes the brain more susceptible to developing mental health issues. Also, through this data, it also suggested estrogen replacement therapy, which can be effective in mitigating some of the mental health symptoms associated with menopause. The most significant findings of this research were the identification of critical life stages, specifically in menopause and postpartum periods, where women experience heightened vulnerability to mental health issues due to the sudden drops in estrogen, because when you're pregnant, you have a high estrogen level, but then after you give birth, it drops sharply. And these findings underscore the importance of monitoring mental health and alongside hormonal treatments for women undergoing significant hormonal shifts. Conclusion. Next, please. In conclusion, the central research question of my thesis was how do fluctuations in estrogen levels throughout a woman's lifespan affect her mental health? Based on the analysis of existing literature, I found that estrogen fluctuations do indeed have a significant impact on women's mental health, with both depression and anxiety showing strong correlations with periods of hormonal change. The contributions of my research in broadening the understanding of hormonal influence on mental health across different life stages by highlighting the vulnerability of women during menopause, pregnancy, and puberty. My work emphasizes the need to target mental health care that considers hormonal fluctuations, offering a broader lens through which we can address women's psychological well-beings. However, it's important to note that the limitations of the study, since this is a secondary literature review, the findings are based on existing research rather than primary data collection. Additionally, the lack of long-term studies in the current literature means that there's a causal relationship between estrogen fluctuation and mental health. Future research should aim to explore these relationships through more long-term studies, as I said before, to provide more definitive answers. Next. Lastly, I would like to thank Gifted Gabber and especially Professor Jen, because she has helped me organize my paper and has supported me through this journey, and also Professor Varkey for giving me more knowledge on neuroscience and providing a strong background for my research. And then an overall thank you to Gifted Gabber and Coach Joe for providing me with this wonderful opportunity.
Wonderful. A question from Professor Varkey.
Job. It was a good presentation. So mainly what you presented is about the lack of estrogen resulting in depression or these kind of issues, especially associated with menopause and all. So, but did you also try to look into this is a problem, what could be a solution? If there's lack of estrogen, did you look into how you can supplement it, or are there any ways, even through diet, such as having? Yeah, please go
I
ahead.
did find one thing that said it's called estrogen replacement therapy, which mitigates some of the mental health symptoms associated with menopause, but I haven't found many other interventions, which is why I think that this research can aim to provide more medications or prescriptions that directly fight the causes of hormonal shifts?
Okay, so one thing that I could suggest is that, sorry, one second. One thing that I could suggest is you could probably try to look more into the role of phytoestrogens. They are like something, which you can actually, these are the different kind of foods that you can incorporate, which can result in estrogen-like activity in our body, and it helps with the mood, or, depression and everything. So it's a very, I know that like in most of the studies, or like the current studies, they don't explore food sometimes, but food can be really a true medication as well. Okay, if taken properly. So anyway, good presentation on it. Good job.
Thank you.
Yeah, I'm
Okay,
also
now we have Professor Jen. Yeah.
yeah,
Yes.
for that, I'm really impressed with both your research and your presentation. You have successfully included everything that was expected in this paper, but you should also follow the suggestion of Professor Varki because it could make your paper better and more presentable. Um, my question is, since you mentioned a few limitations in your paper, how do you think addressing these limitations in future studies could change or maybe enhance the results of your paper?
Yeah. So I mentioned that there wasn't a lot of long-term studies, which kind of limited um my like the information that I could find. And I think that this paper may hopefully it can bring awareness to a broader issue of hormonal shifts in a woman's life. And maybe now that like this is becoming a bigger topic, people will start doing more um like closer analysis on women throughout their life to properly understand how like the proper estrogen fluctuations and the different levels, and to like properly analyze how like specifically women's mood can change. And I think like multiple experiences um and experiments can help like find a more definitive answer to this topic.
Wow, very well said. Congratulations, Tamia.
Thank you so much.
Wonderful. Wonderful. Thank you so much, Amia. Sorry, it's taking this much time. I could have split it, but my team didn't think about it. And by the way, the whole order has been decided by my team. I'm just running through with it. We're almost there. Sunday evening, we'll get through with this. I really appreciate your patience. So let's go with
Also,
Gitanjali.
would it
Yeah,
be
sure.
okay if I leave right now because I have my shift and
Okay,
I'm really
yeah, we agreed on
sorry
that.
about
Okay,
it. Thank you so
Gitanjali.
much.
Yeah, Gitanjali, you want to share? Wonderful. Mhm.
1 second. I think it's. Can you give me a few minutes? I think there's something wrong with
Okay, I will. You want to share your presentation, honey? I can present it for you. I think if it doesn't allow you to present, let's go with Hetwi. Hetwi,
Yeah.
there you are. Yeah, let's go with you. Yes, yes, Hetwi. Are you able to present? If not, you can share the presentation with me. Yeah.
Can you see my screen?
Yes. Yes, I can see that. Go to the first screen and then start presenting, honey. Wait, go to the first one, the first page, the first page. It's in thank you. No, 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 no. Right there, down.
Okay.
It's in
So
thank
this is
you.
first?
Yeah, perfect. And now present and present.
Okay.
There's a present, yes, and present.
Yeah.
Perfect. Go ahead.
So hi, I'm Hetvi Patel, and I am a sophomore in Capel High School in Texas. My research paper was on a multi-model neuroimaging. Wait, hold on, these. Sorry. Okay. Was on multimodal neuroimaging analysis of concussion and subconcussion by identifying a common neural signature.
Okay, so my primary focus is on concussion and subconcussions, which can lead to complex and often subtle changes in the brain functional connectivity. The brain functional connectivity is a synchronization of activity between different brain regions, reflecting on how they coordinate for day-to-day tasks and functions. These injuries impact the prefrontal cortex or the PFC, which is critical for decision making and self regulation. The PFC is also very highly susceptible to concussions and other sorts of TBIs. So my paper asked about the differences in functional brain connectivity and healthy controls versus concussed individuals during their resting state. So to do this, I had to identify subtle changes that may not be visible through standard methods, and I had to determine which imaging technique was the most effective to reveal these subtle changes. The primary objective was to identify structural and functional changes that can be indicative of a common neural signature of a brain injury. For my literature review, there's previous studies and current studies that highlight the impact of concussions and on cognitive and emotional regulation. However, they lack clarity on subtle neural changes. Diffusion-weighted imaging, DWIs, and PET scans are emerging as effective tools for detecting microstructural and metabolic alterations, and recent advancements suggest techniques like DWIs and PET scans can help us see the more subtle changes related to these kind of injuries. So on the top right over here is a DWI scan. This white portion over here is restricted water diffusion, which means that water isn't able to move through the brain tissue as easily as it's supposed to be, which could potentially mean brain tissue damage. On the bottom right is PET scan, and these warmer colors mean glucose metabolism, which mean a high functionality, which is what you want. During concussions, these warmer areas could turn a bit cool toned. So a bit of the weaknesses or limitations on my literature review is on the limited data sets on concussions and the limited sample sizes that I had on specifically on concussions and the inconsistencies in findings, which means that there's still a lot more to explore on this area. As for my methodology and results, I did a statistical analysis from open source datasets that I could find. Data from open source neuroimaging datasets, such as OpenNeuro, were analyzed using Python libraries, focusing on T1 weighted MRI DWI and PET scans. DWI revealed restricted water diffusion in the frontal white matter, showing axonal injury, which is damage to the axons, which are part of the neurons that transmit the signals to other neurons or muscles or glands. Additionally, PET scans showed reduced glucose metabolism in the medial prefrontal cortex. So glucose is the primary fuel needed for neural activity, and decreased glucose metabolism could mean various brain diseases such as cognitive impairment or neurodegenerative diseases. And the findings show that subtle structural and functional impairments were not visible in conventional anatomical imaging. However, these subtle impairments were visible through DWIs or PET scans. So over here is a PET scan on the prefrontal cortex, and reduced glucose metabolism means lowered neural activity, which means low functionality, possibly due to the TBI. For my conclusion, my question was, what are the neural signatures of concussion-related injuries? DWIs can reveal microstructural damages, while PET scans can reveal metabolic dysfunction. However, this study relies heavily on open source datasets, and generalizing these findings to a broader population can be challenging due to the small sample size of the datasets. And thank you for your time. And before I can conclude, I also want to thank Professor Jen and Professor Warkey for their mentorship. They really helped me dive deeper into neuroimaging, and Professor Warkey guided me in interpreting these subtle patterns that I would have missed otherwise. And since I don't have any previous experience in research paper writing or um, things like that, Professor Tubin really helped me with that in terms of organization or just in general, the components that come within research paper writing. Their feedback really helped me strengthen my argument and my paper in overall. And this experience really opened my eyes on brain injuries. And so I want to thank Gifted Gabbers for that. I never knew this topic would be so interesting. So it's like the subtlest thing can be a lot, can matter a lot. So thank you.
Wonderful, wonderful. Thank you for your kind words. You want to stop sharing so that I can add Professor Varkey. Wonderful presentation. Go ahead, Professor Varkey.
Very good presentation. I know it's your first presentation and first research. So that's how the life of a scientist begins somewhere. I believe there is like something that you have started today, which might be very useful for you in the future. Regarding the presentation, it was very good. Overall, like it was clear what you presented. So can you think of some other applications for DWI and PET scan images? Where else it can be applied? And apart from the traumatic brain injuries and concussion?
Maybe brain tumors, possibly before the tumor can spread. You can find a tumor with those, like DWIs, they look for the subtle changes. So maybe before the tumor can spread further, you can try to track those down using DWIs or PET scans.
Okay, because DWI specifically checks the diffusion of water.
Yeah.
Okay, and you know why the diffusion of water, like why it's, what causes that? The difference.
Yeah, the water can't like move through the brain tissue as easily, so the tissue can become dead eventually.
Okay, I mean, just to know, let you know, like, so you should also know why the water cannot move. Okay, it could be because of the capillaries are blocked or something like that. So you can look a little bit deeper into that.
Okay.
But you have done good, Hetty.
Okay, thank you.
Wonderful, wonderful. Professor Jen, a question for her.
Yeah, before asking you a question, yeah, your paper is outstanding, Hetvi, and it's truly impressive that you were able to produce such, a challenging work. That's my opinion. And you also organize your presentation in a very comprehensive manner. So congratulations. And my question is that what motivated you to choose this study? Like what gaps did you identify in the existing research that led you to pursue this topic? Just making your paper unique.
So concussions are really they're really it happens a lot in car crashes or athletics especially. So it's a very major problem. However, not a lot of studies really go in depth about the subtle changes or the neural signature that could show that hey, this is a brain injury.
Wow.
So I just wanted to dive a little bit deeper. I didn't want to go in the broad aspect that most studies really go forward for, but yeah.
Very well said. Thank you for that, Hedvi.
Awesome, awesome. Thank you. Before I forget, there's something important that I want to share. Hold on a minute. Let me just come on the screen. Okay, we know that you have your AP exams in the first week of May. So we have our wonderful study skills coach. By the way, we offer study skills, time management, all those skills. So Coach Anusha is going to conduct 2 sessions on April 13th and April 20th, of which I think I may have sent you the invite invite. If not, please attend her sessions. She's going to teach you strategies as to test taking, test prep, how to deal with stress and everything. So I have a wonderful set of people that I'm very, very proud of. Of course, we have Professor Varkey and Professor Jen, but I also have wonderful counselors and psychometrists who are study skills and time management experts, and a little bit of technique goes a very, very long way. So that's something that we want to offer you. We want you to do exceptionally well. Okay, so great. I got that settled. Let's go with Ketan. Ketan, are you here?
Yes, I'm here.
You want to turn on your video?
Yes.
Okay, great. And Aditi, was it you that couldn't share your screen?
Yeah, hi. I can present now after Ketan.
Okay, so after Ketan, I'll let you go. Otherwise, you can share your presentation and I can present it. Okay? Okay,
Thank
go
you.
ahead, Ketan. Yeah, go ahead, Ketan.
Are you able to see my slides?
Yes, Kaden.
Okay, perfect. Hi, good evening, panel members. My name is Ketan Antoni Pillay. I'm a 16-year-old student at junior attending Mount Olive High School in New Jersey. I'm a Rutgers Allied Health student, and for my HOSA chapter, I am presidential beneficiary. I'm a Taekwondo instructor who instructs for all ages, specializing in special needs care, and I am the officer for Mount Olive's Mental Health Committee. My main topic for my research paper is adolescent gambling disorders, looking through the lens of psychological and social factors. So within psychological and social factors, I was left with a couple main questions that I wanted to answer with my research projects. Why are adolescents in the gambling scene in the first place? Normally, when we perceive gambling, it is usually in the lens of an adult figure. So having adolescents within the same scene made me feel curious, and I wanted to understand why they were present in this moment. I also wanted to understand what effects that can have on them in a social setting. So in terms of strains within a relationship, and also what habits can form from that. My main two objectives was I wanted to evaluate the psychological factors of a gambling disorder, including brain development, impulsivity and conditioning, as well as cognitive distortions that are involved, as well as evaluating the social factors of a gambling disorder in an adolescent through cultural normalization, family influence, and peer influence. So, okay. Today, I wanted to take a moment to talk about what happens when somebody is struggling with a gambling disorder. A gambling disorder in itself is an identified pattern of repeated and ongoing betting and wagering that continues despite creating multiple problems in several areas of an individual's life. At the center of it all is a part of the brain known as the prefrontal cortex, or the PFC for short. This region is responsible for some of our most important functions, making decisions, controlling impulses, and regulating our emotions. However, in individuals with a gambling disorder, the PFC doesn't work out the way it should. Their activity is often reduced, which makes it much harder to resist the impulsive urges to gamble. This reduced activity does not just affect willpower, it disrupts the brain's ability to weigh consequences. Making thoughtful decisions and controlled, risky behavior. Even more importantly, the PFC plays a major role in working with the brain's reward system. In gambling disorders, this connection breaks down when the PFC isn't doing its job properly. It fails to regulate the brain's reward responses. That leads to an excessive risk-taking and compulsive gambling. Dopamine, which is the brain's feel-good chemical, gets released in large bursts during gambling wins or even near misses. And this is because the PFC isn't modulating that response correctly. The brain then starts to associate those intense dopamine surges with gambling. Over time, this repeated stimulation changes the brain's chemistry. Especially, it alters the sensitivity of dopamine receptors, especially the D2 receptors. These changes make it harder for the brain to enjoy natural rewards, meaning that things that used to feel good just don't anymore. Instead, the brain becomes increasingly dependent on gambling to feel that dopamine high. Thus, gambling disorders can be better understood as we're not just talking about poor choices, but we're talking about real physical changes in the brain that can affect behavior, impulse control, and emotional regulation. At the center of it all, as I said before, is the prefrontal cortex. This region is responsible, again, for decision-making, impulse control, and emotion regulation. But in individuals with a gambling disorder, as I said before, it shows a reduced activity. And that reduced activity means it's much harder to resist the urge to gamble. Decision-making becomes flawed, impulse control weakens, and the brain's ability to properly process rewards becomes completely distorted. This distortion happens because the PFC doesn't act alone. It closely interacts with the brain's reward system. In those with a gambling disorder, this connection becomes unstable. The weakened inhibitory control of the PSC allows for excessive risk-taking, leading to compulsive gambling. This state even near misses the moment where someone almost wins triggers intense bursts of dopamine, or actual wins. Dopamine cues trigger dopamine releases through the metabolic pathway, from the VTA to the NA, creating an intense motivational drive to keep gambling, no matter the consequences, which then creates pathological gamblers. Within the prefrontal cortex, there are three major areas that are at play. The orbitocortex, abbreviated to the OFC, evaluates rewards and punishment. The dorsolateral prefrontal cortex, the DLPFC, supports working memory and cognitive control. And the ventral medial PFC, the VMPFC, manages emotions and decision-making. In gambling disorders, the DLPFC shows hyperactivity, reducing control over impulsive actions. Meanwhile, the VMPFC shows hyperactivity, increasing emotional reactivity and reward seeking. This imbalance means that logical thinking takes a backseat and compulsive behavior takes over. And it doesn't just stop there. The longer a person gambles, the more their neurons become tolerant to the dopamine. The receptors at the receiving end begin to shut down, forcing the individual to gamble more to feel the same pleasure. And those near misses that happen, the brain reacts to those near misses almost in the same way as if the person were to experience an actual win. Why is that? It's because the insula, a region responsible to emotional interpretation, misreads these near misses as a sign of a big win and it being close, which reinforces that continued gambling. Other regions that interact with this process are the ventral striatum, which releases dopamine during wins or even anticipates the wins. The amygdala, which heightens emotional responses, amplifying both the thrill of a win and the sting of a loss. The insula, which fuels cravings and false beliefs, like the gambler's fallacy, which is a theory that I will go over later, and the dorsal striatum, which helps transform repeated behavior into a habit, which sets in the gambling cycle into place. So it means that a gambling disorder isn't simply just a matter of bad choices or weak willpower. It's a neurobiological condition, one where critical systems that regulate emotion, decision-making, and reward are fundamentally altered. These changes, especially when they occur during the fragile period of adolescence, can shape behavior for years to come and affect adulthood. The prefrontal cortex, the part of the brain that we just talked about, doesn't fully mature until around the age of 25 years. That means that during adolescence, a time where people are most likely to experiment and take risks, the brain's decision-making center is very much under construction. According to a study by Aryan and colleagues in Neuropsychiatric Diseases and Treatment, adolescence is a critical period of brain development. During this stage, the limbic system, responsible for processing emotions and rewards, develops much earlier than the prefrontal cortex. This creates a gap. Teens experience emotion and reward intensely, but they don't yet have the cognitive control to properly regulate those experiences. This imbalance leads to heightened impulsivity and risk-taking. In the context of gambling, It means that adolescents are especially vulnerable. They chase that dopamine rush, the excitement of a win, or even a near win, and their brains start forming, again, those powerful associations. This practice of creating associations is called brain conditioning. Through two types of conditioning, through classical and operant conditioning, their experiences reinforce gambling as a source of pleasure. Even the near misses, where they almost win, can light up their brain's reward system, making them want to play again. Over time, this repeated stimulation, as I said before, leads to dopamine tolerance. Just like with a substance use disorder, the brain needs more intense stimulation to achieve that same thrill. So teens may gamble more frequently or take bigger risks in an attempt to recreate that original high. By a study by Van de Bos, referenced by Steinberg and Chain, explored adolescents' tendency to act impulsively. Their findings suggest that it's not just about craving rewards, it's also about their cognitive control mechanisms, like those in the prefrontal cortex that are still underdeveloped. So it's a double impact. Their desire for instant gratification is strong, and their ability to resist that desire is weak. This creates a perfect storm for developing harmful behaviors such as compulsive gambling. Now, adding on to that mix is what is called gambler's fallacy. The belief that if something happens more frequently than normal during a given period, it happens less frequently in the future or vice versa. For example, if someone flips a coin and gets heads 5 times in a row, they might believe tails is due. But in reality, each coin flip.
Ethan,
Yes.
Sorry, I think your slide is not moving. Is that what Professor Varkey you want to say?
Yes.
Your slide is in as adolescent brain vulnerability. Is that where you wanted it to be?
I'm so sorry. Let me fix that.
Yeah. I paid attention this time.
Would it be more beneficial if I restarted or just continued?
No, you can continue.
Okay. So now add to this mix the way that adolescents think about probability. Yes.
Sorry, it's still on the same slide.
Yes, this is the slide that I want to stay at.
It says adolescent brain vulnerability. That's the slide?
Yes.
Okay, because it seems like it was the second slide that you. Oh no, never mind.
Okay.
It looked like very similar. So
The fallacy is dangerous in gambling because it leads to poor decision making. People believe that the previous outcomes somehow guarantee a certain result next time, even when there's no logical connection. This ties to another psychological error called the law of small numbers, our tendency to take small samples of data and then use them to make big assumptions about the whole picture. Psychologist Amos Tversky and Daniel Kahneman called this insensitivity to sample size. As the American board explains, events can either be independent or dependent. In gambling, most of the time, outcomes are independent. For instance, if you roll a die or spin a roulette wheel, the result is not just influenced by what happened before, but the brain, especially an adolescent brain, often misinterprets these events, thinking that somehow they're linked. This flawed perception contributes to the continued risk-taking, as a gambler believes that they can predict or beat the odds. But in reality, they're making decisions based off of false logic, fueled by underdeveloped brain emotional impulses, and distorted sense of probability.
Okay. Do you want to change your slide or do you want to be on it?
It's not changing.
Okay. Do you want to unbig this unpresent?
Yeah, I think that might be better.
Click somewhere, click anywhere, and it may just move. No? Escape. Click escape. Okay. Now change the slide here. Okay. Yeah, we went through that. Okay, the next one. Yeah. Social factors, is it? Just continue this. This is looking good. You don't need to make it full slide. We can see it well.
Okay, perfect. Okay, so within social factors, adolescents are deeply shaped by the behaviors of those around them, especially their peers. In many cases, gambling is first introduced in casual social settings. Think about family poker nights or fantasy leagues or betting in sports games, scratch off tickets, or even esports betting. These all may seem harmless or just for fun, but for a developing brain, such as an adolescent brain, especially one that's already wired for impulsivity and dopamine-driven reward seeking, these experiences can quickly turn into something much more serious. Social gambling has a weird way of reinforcing itself. When an adolescent wins, even just once, twice, or a couple times, it creates a very strong association. So that gambling can equal success, excitement, or even social status within a friend group. That single win can be enough to plant the seed of compulsive behavior, especially if it's celebrated by peers or seen as impressive. But peers aren't necessarily the only influence that exists. Family also plays a huge role in it as well. Let's take the example if an adolescent grows up in a household where gambling is present. Whether that's a parent who bets on sports or a sibling who plays online poker, it can begin to feel normalized or even aspirational. They might start to view gambling not as risky, but as a regular part of adult life. Sometimes parents unknowingly reinforce these behaviors. Maybe they let their child place a bet just for fun, or they talk about gambling wins without ever mentioning the losses. Even common language, such as "I bet you can't do this," plants gambling concepts in everyday conversation. Over time, these seemingly small actions can normalize gambling in adolescent minds. In other cases, if parents are neglectful, overly permissive, or disengaged, teens may turn to gambling as an emotional outlet. That rush of dopamine that they get from a gambling win can feel like a temporary escape from stress, loneliness, or even a lack of support. There's also a cultural factor. Especially in today's media, social media, since we have a very social media driven world, modern media often portrays gambling as something to be glamorous, fun, even luxurious. Celebrities, influencers, and athletes are openly seeing betting on games or promoting online casinos. Social media, in particular, makes these behaviors seem exciting and normal, especially when influencers post huge videos about huge wins, flashy lifestyles, or gateways that are tied into gambling. And especially for adolescents that are still trying to figure out their identity, it's a powerful influence. If their personality leads towards risk taking and they see friends or their favorite influencers, the temptation becomes even stronger. And for dopamine seeking teens, those images of fast cash and instant rewards can be almost irresistible. So when we look at the bigger picture, neurobiology, peer pressure, family exposure, and cultural messaging, it becomes clear that adolescent gambling is not just a behavioral issue. It's the perfect storm for psychological and social and environmental factors colliding together at the most vulnerable stage of brain development. Now, beyond the social and psychological factors, we also have to consider the serious emotional consequences of gambling disorders, especially in adolescence. Gambling addiction does not just stay at the table or on a phone screen, it seeps like vines into every corner of a person's life. Financial problems, broken relationships, and the heavy emotional toll of guilt and shame are just a few of the outcomes. And when these burdens fall on the shoulders of a teenager, someone who is still learning how to manage those emotions and stress, it hits even harder. Unlike adults, adolescents have yet to develop their full coping mechanisms that are needed to navigate serious emotional struggles. So when depression sets in, it's fueled by repeated losses, failed attempts to stop gambling, or the overwhelming sense of chasing money they can't win back. It's unbearable. The hopelessness becomes consuming for them. The idea that a win is just around the corner, especially under the grip of the principle we spoke on earlier, the gambler's fallacy, builds a sense of false hope. And when those hopes are shattered, that fall is much more painful. This cycle not only just creates depression, but deep emotional despair. Teens begin to isolate themselves, cutting off friends and family, either out of shame or the desire to hide their behavior. But for adolescents, this isolation is particularly dangerous. This is the time in their life where they're trying to build their identity, to figure out who they are and where they belong. So when they feel disconnected, lost, and unsupported, the emotional impact can be devastating and detrimental. Anxiety also plays a huge role in this. Constantly worrying about money, hiding gambling habits, and feeling the pressure to keep playing in the hopes of turning things around, all of these fuel high levels of stress. The unpredictability of gambling leads to emotional chaos. Feelings of restlessness, irritability, and agitation are all common, especially if the adolescent is trying to cut back or stop altogether. And unfortunately, gambling disorders rarely exist within isolation. Many adolescents who struggle with gambling also deal with other mental health conditions, from substance abuse and impulse control disorders to ADHD. In some extreme cases, emotional distress can lead to, unfortunately, suicidal thoughts and behaviors. When an adolescent feels trapped emotionally, financially, and socially, suicide may seem to be the only way for them to find escape. This is a harsh reality, but one that is important to talk about because every statistic is a young person, someone who is unfortunately on a different path. If only the right support, awareness, and interventions were in place, we can prevent that. Some ways for prevention is through projects such as this, showing the awareness and bringing to people's attention the dangers of gambling behaviors within adolescence and teaching people how to recognize these different risk factors that might be presented within an adolescent. Also, having an open support group, whether that's with friends, family, teachers, professors, etc. People that a person can go ahead and talk to and communicate with is also very important. Treatments such as cognitive behavioral therapy and support groups are also very key in order to treat pre-existing signs, as well as policies. So promoting age restrictions and advertising limits to help prevent adolescents from being exposed to such things. For my methodology and results, I utilize credible sources such as the National Institute of Health website to acquire data and support my prompt and analyze the intricacy of a gambling disorder, analyzing the data to evaluate the impacts of a gambling disorder. The main result that I have acquired is that gambling disorders have a huge psychological and social effect in terms of consequences on brain development, mental health, relationships, and behavior. Gambling disorders in adolescents can also create dangerous intersections of neurodevelopmental vulnerability, emotional stress, and social disconnection, often leading to long-term psychological harm and dysfunction, behavioral patterns that persist even till adulthood. So those habits keep continuing. For contributions and limitations, my contributions to the study of gambling disorders is I want to share awareness of gambling behaviors within the adolescent population. Recognizing something that I said earlier about how we usually associate the concept of gambling with the adult age group, it isn't just that. Some of the behaviors can also stem from the adolescent age, which is what this project or research is all about. So it's understanding that we can't isolate just the adult population. And when we're identifying the root causation of most of these diseases, in terms of gambling diseases, it's key to recognize that some items can, again, stem from not only past experiences, but traumatic experiences, which is in the case for my research in gambling disorders. By analyzing that causation in a way that highlights areas of the effect for adult generations, we can gather more profound understanding of the topic at hand and understand how we can further benefit our adolescent population for future generations to come. Some of the limitations that I experienced were in some of the literary articles that I have reviewed. Most of them are based off of input that is given by a patient. So self-report bias, whether they feel that an answer they would want to share to the fullest truth or the fullest honesty will always be an issue or a limitation, because we can't guarantee 100% that they're going to answer with honesty. And secondarily, it's all the resources that I've collected have been from online sources, so none of the experimentation I've done myself. And finally, thank you so much for listening, all of my peers and Gifted Kabar's neuroscience research course. I appreciate the amount of time that we've spent together.
Yeah, thank you so much. I'm going to have Professor Varkey speak, but I see the effects of it here, and my husband is not even an adolescent, so a lot of it resonates with that. Yeah, it feels like this whole presentation is about Coach Joe's life, but I'll let Dr. Varkey ask you questions.
The comprehensive presentation, Ketan, like it included quite a bit of information, and I see that you have gone quite deeper into your research area. Very good. Just like so, question. So, for those who are addicted to gambling, what kind of treatment is available? Have you included that in your research, or are you planning to include that?
So, in terms of treatment for those that are experiencing a gambling disorder, it's always going to be somewhere, it varies depending on how extreme that gambling disorder is. So, whether it's towards the far end of the extreme, so if they're becoming a danger to themselves or other people, maybe that might be admission into either a psychiatric unit if it becomes to that extent, or something as more mundane as therapy with a licensed therapist or a clinician.
Yeah. Can the particular like brain region be be affected, like the production of dopamine in some way using some kind of non-invasive techniques?
So, sorry, could you restate your question, please?
By using certain non-invasive techniques, like magnetic stimulation and all, magnetic stimulation, do you think that it's possible to influence the gambling addiction for an individual?
Based off of the research that I've conducted, I believe that it would depend on the stage and the duration of how long that gambling disorder has resided for. If the gambling disorder has been in the person's like prevalent or has been pre-existing the person for a while now, for let's take a couple years, it might be difficult for that to be a viable treatment option because the neurotransmitters that in the receptor, it will already have become desensitized to that dopamine rush. So even if you use different tactics to decrease the amount of dopamine within the body, it might not affect as much for somebody that has been experiencing a gambling disorder for a long time, but it might be a viable option for those who have or are experiencing gambling disorder for a very short period of time. But you'd also have to take into consideration that when reducing dopamine levels, it might not only impact the gambling disorder itself, but it could also impact the way that the person regulates their own emotions. So if it impacts their personality, how they feel joy, etc.
Good answer, Ketan. Good job.
Okay, we'll have Professor Jen ask you a question.
Hi, Kaden. You know what? This paper shows great promise and has really the potential to make a significant impact, especially you know, it addresses the pressing issue nowadays, gambling among adolescents. You know, it's very alarming. So thank you so much for coming up with this paper. And I can say that your work is thoroughly, I mean, uh, yeah, it's a thorough and well presented, organized as well. You were able to demonstrate a strong grasp of the topic. So congratulations on that. Now, my question is that um if you were given more time, more resources, what areas of your research would you like to explore further? And like, how do you plan to build on the conclusions you have drawn?
If I were to have more time for this research project, I believe that I would have looked through the issue of adolescent gambling through more lenses than just the psychological and social one. Because even though they are the two main pillars that I decided to focus on, gambling disorder and the human brain has such a wide range of topics that we can go off of. I would be very interested to delve in even more onto what that is. And secondarily, as aside from your question, I did also want to thank Dr. Varkey as well as yourself, Professor Stephen, for the amount of work that you've put in to aiding me in this project. The gambling paper and sorry, the paper that I've written, as well as the experience with Gifted Gabar, has been very enlightening for me. I've loved working with both of you. It has been an amazing experience, and I appreciate Coach Joe as well. Her expertise on how to formulate this, like the program itself, I enjoyed it thoroughly. So thank you so much for that.
Yeah, thank you. Yes, this has been a good presentation. And also, I'm part of another organization where we build awareness to different youth issues, and I will be inviting you there to speak about this, right? It's pretty good and quite enlightening. Thank you so much for sharing your work with us. Okay, so I'm going with Prashana. Prisha, are you there? Oh, it was Prisha, right? I'm sorry, I'm losing my mind. Prisha,
Yeah,
are you there? Yeah.
yeah, I'm here.
Okay, you want to turn on your video?
Yeah.
Okay, great.
Okay.
Yes, I'm just going to quickly share my screen
Oh,
and
it's
oh,
working.
you're able to share? Okay, great. Yeah, present and present. Yeah. No, present. Correct. Present. Yes.
Yeah. Okay. Can you see my screen?
Yes.
Okay. Hi everyone. My name is Prashant Charji. I go to Woodbridge Academy Magnet School in New Jersey, and my topic is monoclonal antibody therapies for Alzheimer's disease. This is my thesis defense. Oh, sorry. Okay. So again, my topic of investigation is the MABs for Alzheimer's disease. MABs stand for monoclonal antibody therapies. And my research question in particular is, what are the advancements and challenges of monoclonal antibody therapies for Alzheimer's disease? This question is crucial because monoclonal antibody therapies they represent a promising avenue for treating Alzheimer's disease, which is a very well-known condition that has long lacked effective treatments. And it's actually very interesting to note that it is the number one neurodegenerative disease with over 60 to 80% of dementia cases all falling under Alzheimer's. My research objectives for this are to critically assess the current advancements and challenges, and to evaluate the clinical efficacy, safety profiles, and the potential of these therapies to slow cognitive decline in patients with early stage Alzheimer's. So for my literature review, I used many secondary sources, but one of the main things I did was kind of give a background and overview of the disease itself. So as you all know, Alzheimer's falls under the umbrella term of dementia. But what you may not know is that the disease is characterized by the accumulation of amyloid beta plaques in the brain. And I'll kind of go back a little bit with this. So amyloid beta monomers they aggregate or they accumulate to form oligomers. And by that, I mean like they cluster up. Then the oligomers cluster up to form protofibrils and fibrils, which are kind of this insoluble thread-like looking thing. And then these in turn cluster up to form plaques, which are so you can think of the protofibrils, fibrils, oligomers as building blocks that lead up to the plaques. So by the accumulation of amyloid beta plaques in the brain, which are believed to disrupt communication between the neurons, or like cells, and lead to neuronal damage, which ultimately causes the cognitive decline and memory loss that we see in typical Alzheimer's patients. The pathogenesis of Alzheimer's, or the kind of cause, is largely understood through the framework of the amyloid hypothesis, which kind of states that due to this accumulation, that is kind of the central driver of neurodegeneration in Alzheimer's disease. So here, if you can kind of see, we have a healthy brain, and this is an Alzheimer's brain where we can see plaques, accumulation of amyloid beta plaques, and that's kind of that due to that buildup that's causing the cognitive decline. And if anything, you can see the brain, the size of the brain in comparison, and the shrinking. Then two types of monoclonal antibody therapies that I kind of looked into were aducanumab and lecanemab. So the first FDA-approved MAB, or again monoclonal antibody therapy for Alzheimer's, was aducanumab, which actually received approval in 2021, which is fairly recent, despite considerable controversy, which I will discuss a little later. So aducanumab is a human monoclonal antibody that selectively binds to aggregated amyloid beta plaques, promoting their clearance via microglia activation. So what that means is you can kind of think of microglia as the cleanup crew cell in the brain. So by binding to these amyloid beta plaques, this antibody aducanumab kind of sends a signal that helps clear up the plaque damage. Furthermore, for lecanemab, it targets soluble amyloid beta oligomers, which again are actually believed to be more neurotoxic than the amyloid plaques themselves. So again, the oligomers are kind of one of the building blocks of the plaques. So one of the trials that was looked into in this study, in this research paper, was the Clarity 80 trial, which demonstrated that lecanemab was able to reduce amyloid plaque burden and slow cognitive decline by 27% in patients with early stage Alzheimer's disease compared to the placebo group, or the group that didn't receive this therapy. So for my methodology and results, I used secondary sources like peer-reviewed journal articles, clinical trial reports, as mentioned before, and research papers that are already discussed within the paper. And these sources also include reviews and articles from journals, as well as clinical trials like the Emerge, Engage, and Clarity AD trials. So the results from various clinical trials of monoclonal antibody therapies have been a mix of success and failure, actually, which I guess is kind of expected because Alzheimer's is, of course, not just one simple, straightforward disease. There's so much to it. There's so much that goes behind it. But the Emerge and Engage trials, which were studies designed for aducanumab, also showed mixed results in particular. And they had it was associated with ARIA, which I will also talk about more on the next slide. And these concerns raised alarms about the safety of the drug, especially in patients carrying the APOE E4 allele, which is just a genetic risk factor for Alzheimer's disease, but nonetheless makes it especially concerning. The Clarity AD trial, which was more centralized for lecanemab, demonstrated a little bit more consistency. And in this study, lecanemab, like I mentioned before, showed a 27% slower decline in cognitive function compared to the placebo. But lecanemab was also still found to be associated with ARIA, which over here, I'm talking about more. It's amyloid-related imaging abnormalities. So, this was observed in a significant number of patients, so both aducanumab and lecanemab was associated with this, even though they both had their respective pros and cons. And this raised a lot of concerns and controversy about the approval of the drug by the FDA, especially because, again, there is so much ambiguity with it. So, and to the general public, this had a lot of concerns and issues raised. Lecanemab was also found to be linked with ARIA, and it's not just the amyloid-related imaging abnormalities, it's also microhemorrhages, brain swelling, etc. for both. But again, just overall, due to the 27% slower decline, I understand it, I mentioned it a few times, but I think that was one of the key factors in the sense that lecanemab kind of had that as compared to aducanumab, which didn't. And these findings overall suggested that due to this, lecanemab could have a more reliable effect on slowing this cognitive decline in early Alzheimer's disease compared to aducanumab. So to restate my research question again, so it was what are the advancements and challenges of monoclonal antibody therapies for Alzheimer's disease? Some limitations that I had when coming across this were the side effects, of course, because this was a big deal when you're administering a drug to a patient and you don't even know if it has a half chance of making their condition better or if anything worsening it. So ARIA, again, amyloid-related imaging abnormalities was a big part of this, and the clinical benefit that's seen in early stage patients doesn't necessarily translate to long-term improvements, which is also something that I found as a limitation because just because it works once doesn't mean that it'll work for the duration of their disease, if anything. Um, as for my contributions, I utilized the secondary sources, the journals, the articles, and kind of understood how to link the causes of the Alzheimer's disease with the alpha beta plaques buildup and how monoclonal antibody therapies kind of bind and work and function in relation to this. And when I say challenges and advancements, you can kind of think of it as uh, pros and cons of the therapies. My overall research experience, I want to give a big thank you to Dr. Varkey, Coach Jen, and just Gift and Gabber for helping me and supporting me through this entire research process. Um, if anything, Dr. Varkey helped me a lot with understanding the particular how the nuances and how everything works because there was a lot of terms and things that I didn't know. For example, Dr. Varkey kind of explained this to me yesterday, but he said that you can kind of think of amyloid beta plaques as kind of the villain or the evil threat in the story. And that kind of made a lot more sense to me. And I think I was also, because I was able to understand that, I think that I was also able to effectively learn and communicate that here as well. But through this, I also learned how to effectively communicate, not just in my paper, but also while creating this presentation. And overall, with Coach Jen, she helped me improve my research skills and formatting skills. And I learned how to analyze data and ask the right questions in terms of how to evaluate findings. So I think that was a big thing that made my research experience unique and very beneficial and educational. So yeah, thank you.
Wonderful. Professor Jobin, a question for you. Yeah.
Thank you, Prisha. It was a very good presentation, clear presentation. One question for you, Prisha. Why do you think, or what are the challenges in, like developing a antibody or a monoclonal antibody in Alzheimer's disease?
I think because well because it's also I mean because there's a lot of like the plaque buildup, it's not just one particular thing. There's a lot that goes behind it in the sense that like how I was talking about the it's not alpha, amyloid beta, plaque buildup, the monomers, fibrils, oligomers. I think pinpointing exactly where that buildup is because the brain is very complex. It's not like you can just attack one region and that's that. It's all linked. So I think the binding is one thing, but also kind of pinpointing how that works in relation to other parts of the brain could be a challenge when creating a drug, especially because um, it's not like it's visible, it's administered intravenously. So
It's what like I was looking for is like when you talk about the monomers, oligomers, and fibrils, remember that these oligomers, as we talked about like a couple of days ago, that they can be of different conformation, the different shapes, okay? Different shape and different sizes.
and
And
mutations.
these shapes and these sizes, yeah, and they vary from patient to patient. So eventually it is very difficult since the target itself is different. As you now remember from the story, the villain itself changes. We need
Yeah.
to have a different like method to target that. Good job, Prisha.
Thank you.
Wonderful. I mean, I'm not going to stop any student today, but a lot of you guys have gone off script, which is perfectly fine. I love to see that you all want to come here and express. It's wonderful. So yeah, let's go with, wait, Aditi? Aditi. Oh yeah, I'm sorry. Professor Jen didn't ask a question. I apologize. I'm losing my
It's
mind.
okay. It's okay, Coach Joe. Yeah. Prisha, you have done an impressive job in presenting the advancement and the challenges about this topic. I'm just curious to hear more about your approach, especially, writing this paper in terms of how you tackled the vast amount of literature on the topic. So, writing your paper, how did you ensure that you presented a balanced view of the advancement and then the challenges, particularly when dealing with conflicting perspective in the existing literature?
Right. So, that's a good question. I think because there was just so much information to process, I kind of kept the two things in mind, advancements and challenges. And when I was reading the different journals and articles, first I tried to understand what the entire thing was about. What are monoclonal antibody therapies? What is amyloid beta plaques? What is APP, amyloid precursor protein? What is all of that to begin with? And then I kind of focused on the interesting aspects of the advancements of challenge and challenges for each particular type of drug. And I also saw that there were many others, but just based on what I'd seen in the different papers, these two, aducanumab and this lecanemab, were the two most mentioned ones.
Wow, impressive. Congratulations.
Yeah, wonderful. I mean, sometimes I have to pinch myself to tell myself that these are just high school kids. Okay, so amazing, amazing. Let's go with Aditi. Great job, Prisha. Very eloquent, very nice, impressive. Okay, Aditi, did I lose you?
Can you hear me?
Yes, I can.
Can you see my screen?
Yes, I can. But I don't see you. You want to turn on your video?
Oh, it shows that my video is on. I'm doing my video on the not on the app. I'm doing it on the web page.
Okay, but we see it chopped off. Okay, it's like kind of chopped off. It's not complete.
Um,
You want to share your presentation to me and I'll present.
yeah.
I can have someone else go by then. Okay,
Okay, thank you.
no problem. Yeah, Kinjal, you want to go, honey? Kinjal, you there, or did I lose her? Did I lose Kinjal? Kinjal Singh,
Oh,
you
yeah,
there?
sorry.
Hi, sorry, it's been a long day and I apologize for the
It's
delay.
okay.
You want to, yeah, turn on your video, honey?
Yeah, sure.
Great. Okay, there you go. Okay.
Okay. Yeah, I should share my screen, right?
Yes, absolutely.
Yeah, cool. Mm. Okay. So this is my presentation on the neuroarchitecture of moral divergence. And I did this because I was inspired by um like the question and the factor of why good people do bad things in terms of looking at like serial killers and psychopaths, and I really wanted to understand the activity that was going on in their brains when committing these crimes. And my topic explores the areas of cognitive development and neuroscience. The questions I had and wanted to answer were like, how do ordinary people commit to unordinary acts and unethical acts, and what differences in brain activation occur when individuals make decisions that benefit themselves at the cost of others. And what I really wanted to do was to understand the 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 developmental activity going on in the minds of these people, and how the the mapping of their brain
led to their actions. And I use both primary, and secondary resources to better reinforce and analyze my key questions. And I analyze I analyzed like forensic insights in the context of brain abnormalities, and then compared the MRI scans of both criminals and non-criminals, and I used neuroimaging studies in criminal behavior and focal brain lesions. The methodology and the results I had was I analyzed recurring patterns in brain activation and looked for like activities the in like each regions of the brain in hopes of finding something that played into what I found, but of like what made the like criminals act this way. And I also assess impairments and structural changes in the brain and how they influence decision making and empathy. And people with abnormalities in their structural brain in regions such as the prefrontal, anterior, and ciliary cortex are more likely to engage in unethical acts or to reduce moral judgment capabilities. And abnormal activation in the prefrontal cortex is associated with impairing moral reasoning and impulse control, directly influencing decisions that benefit individuals at the expense of others. Some limitations that occurred when researching was normally the research I did like come across was more about like psychology instead of actual neuroscience, and it didn't really have much information on the brain rather more of the behavioral aspect of it, which I get because it was more of like this is more of a psychological factor like rather than like a neurological factor. But then again, I wonder how like thoughts that occur in the brain like did like originate in the first place, and it's not just because of psychological factors, it's also about how the brain functions too. And and then when I looked at some case studies regarding some criminals, they were often like based on anecdotal evidence from the criminal itself, which is pretty biased in terms of how does their mind work, because it's only coming from them and not another outside force. And when you're looking at like neurocriminology, it's only like there are only studies done on small like sample sizes of like a research group. And so we don't, and that like kind of affects like the validity of the research because it's not like a whole wide span. It's just a small group of people, and that doesn't have a say on the entire population. Thank you.
Okay, you want to share your experience about the program?
Mm-hmm.
Yeah.
So I was like having trouble with this research paper because I signed up at a time when I was going through a lot of stuff in terms of extracurriculars, and I was like doing a few other side passion projects on the side, so I really didn't have the time and like the effort and the energy to initially like start my research paper, but people like Professor Tubon like really helped me try to like regain my footing a little bit, and that was really grateful, and I was really grateful for their patience.
Wonderful. Great. Thank you for the experience. I'll have Professor Warki ask his question.
Kinjal, like it was very interesting presentation.
Mm-hmm.
Although I always saw that you tried to book, but then book and cancel.
Yeah,
all
I'm
the
so
ones.
sorry.
So I think like I would have been happy to help you
Mm-hmm.
before this, like before presenting this.
Yeah.
Yeah, this topic is like, of course, like it's very difficult to predict just based upon a brain scan whether
Mm-hmm.
an individual is going to be or he has like criminal tendencies or not. Definitely, we can just say that there are brain abnormalities, but
Yeah.
like that could be even for people who, I mean, it could be due to Alzheimer's, Parkinson's, anxiety, depression. There's so many
Mm-hmm.
different things going on in the brain. So, I mean, I don't think that it's something that can be done yet, at least like to my knowledge of neuroscience, whatever I have. I don't think that we are yet to, like decide based upon a brain scan whether individual can have these tendencies. That's more like a psychological analysis, and which can change from time to time, of course.
Yeah.
So anyway, like yeah, keep working, trying to maybe like modify a topic a little bit, change it, and then maybe you'll find something more interesting to add to this study what you have started. So it could be more from a psychological perspective instead of from a neuroscience, like perspective.
Yeah.
Okay.
Okay. Now, Professor Tuban, question for her.
Hi there, Kinjal. Your presentation clearly demonstrated your understanding of the topic despite your busy schedules and prior commitments. But I'm just impressed. And, continue to work for your paper. This is like, I mean, you have to work on this. This is the gateway for you to be surrounded by different opportunities, bigger opportunities in the future. Anyway, I just have to ask you one question just to confirm, like, what strategies did you use to synthesize the vast amount of information you gathered from different studies, and you able for you to be able to create a cohesive narrative that aligned or tied back to your research question?
Normally, okay. So, what was the biggest like problem with the topic I did in general? That it was more of like psychological and behavioral based instead of like neurological based. And so I tried to go back and look at like MRI scans of like old serial killers and try to like compare them and research like the neurological activity going on in their heads and see if that had any factors in terms of their behavior and how like they committed these crimes. And it was more or less like there was just like active it was just like anomalies in places like the prefrontal cortex and the amygdala, which like inhibits empathy and decision making. And so that's what I mostly looked into.
Wow,
And
impressive.
it tied in with like the psychological aspect of it, like, oh, um, they didn't really have any ethical concerns in terms of what they were doing. So yeah.
Very well said. Thank you for that.
Very interesting, Kinjal. I mean, the range of research topics we've seen today is pretty amazing. Thank you for being so patient today.
Yeah.
Let's go with Prisha. Prisha, your presentation is working. Let me just share my screen. Okay. Sorry, it was Aditi, right? I'm losing. Is it Aditi?
Yeah.
Okay. Aditi, let's just get started. You can present, and I'm going to share the screen. Okay, go ahead with your presentation, and I'll start sharing.
Hi, my name is DT Gosavi. I'm a freshman in Newport High School, and this is my presentation about neuroplasticity. Can you change the slide, please? So my goals in this research is to explain what neuroplasticity is and get more understanding, examine its role in learning and memory, and analyze neuroplasticity's importance in stroke rehabilitation. So for my literature review, I just use databases like Google Scholar, ResearchGate, and PubMed with emphasis on studies within the last 10 years. So I also used research MRIs and research articles and review papers, which helped me showcase a variety of perspectives and analysis. So, what is neuroplasticity? Neuroplasticity is a really interesting topic that allows the brain to adapt, reorganize, and form new connections. It plays a really crucial role in various processes like learning and memory formation and recovery from brain injuries like strokes. In this paper, I'm going to explore three main areas: the mechanisms behind neuroplasticity and its applications. So, neuroplasticity involves adaptive changes in both the structure and the function of the brain. These changes can occur at various levels, and from strengthening synaptic connections to forming entirely new neural pathways. One important mechanism of neuroplasticity is synaptic plasticity, where the synapses, which are the connections between the neurons, change in strength depending on activity levels. So, this process is further explained by something called Hebbian learning, which suggests that neurons that fire together are like wired together. And this principle helps explain how experiences strengthen connections in our brain, turning short term memories into long term ones. Neuroplasticity also involves structural changes in areas like the hippocampus, which is key for memory, and motor cortex, which controls movement. However, I would like to add that um, we do not actually like know where memory is in the brain. So, yeah. Okay, another thing is structural plasticity, which is a really important key mechanism, which involves the creation of new neurons, which is known as neurogenesis. And this is also this also occurs in regions like the hippocampus, which is like really crucial to memory formation. So I'm going to get into my first topic, applications of neuroplasticity in learning and memory. So neuroplasticity plays a vital role in learning and memory, and there are many studies for this, which have shown that practical skills like juggling can increase gray matter in the visual cortex, as well as other brain regions involved in motor coordination. In medical students, significant changes have been observed in the hippocampus and parietal cortex, which are involved in memory and sensory processing. Neuroplasticity is also evident in musicians and bilingual individuals, which is where we saw that increased brain connectivity in areas such as the prefrontal cortex, improved skills in auditory processing and motor coordination. Additionally, cognitive exercises like listening to melodies can optimize learning by triggering brain activation. So, neuroplasticity is really cool because it's also crucial for recovery. So, when the brain goes through injuries like a stroke, the brain is trying to compensate for damaged areas by reorganizing neural pathways, which is like kind of like rewiring itself. And research shows that therapies like constraint induced movement therapy, CIMT, and transcranial magnetic stimulation, TMS, can stimulate this plasticity to help regain lost motor functions. Studies by researchers like R.J. Nudo have demonstrated that such therapies promote motor recovery by encouraging the brain to engage undamaged areas. So, in conclusion, neuroplasticity is a crucial mechanism that shapes how we learn, recover, and adapt throughout life. It has profound applications in fields like education and stroke rehabilitation. Yeah. Thank you so much. I want to thank Professor Varkey and Professor Jen for supporting me. This is my first research paper that I have like kind of ever done. And I had a lot of fun doing this process, and I really appreciate the weekly meetings that helped me understand more about this research process because like it's really complicated and I didn't know what things like an abstract was before this process, and I really appreciated the amount of time it took for these meetings. I'd also like to thank Gifted Gabber and Coach Joe because this is like a really cool opportunity for me. I've like never done things like this, so thank you so much.
Wonderful. Great job, Aditi. Dr. Jobin, question for Aditi.
Good job, Aditi. Like for the last talk that we had, I see that you have added like quite a bit of information. And I know that it's since your first presentation, but you have done a good job. So my question to you regarding this topic is like, have you looked into I know that you mentioned that like neuroplasticity is important with respect to stroke and everything. But have you tried to include the treatment options or like how neuroplasticity can be regained in your actual research study?
I'm sorry, I can't hear you.
I don't know. Yeah, I have no idea why it's happening though today. Can you hear now?
Oh yeah, it's a little better.
Like maybe something with my, although I'm talking with my AirPods on, still for some reason, maybe some connectivity issue. My question to you was, have you tried to add information regarding the improvement of neuroplasticity or treatment, or like how we can gain uh, functions such as in after stroke? What are the different techniques used to treat stroke?
I'm sorry. Can you type it in the
You
chat?
can't hear?
No.
Okay, I'll put it in the chat. Mine. You probably, Professor Jen, you can go ahead if you can hear me.
Yes, Professor Joven, thank you
Is
for
it
that.
even you're not able to hear me clearly? Is it?
Yeah, it's like you're too far from your unit. I think even if you have the headset, I think you need to change like the setting. You have to choose like a device
Yeah.
that needs to be connected. So it seems like
No,
you're
it's
just using
connected.
the speaker. What about now? I think
Okay.
it's better
Okay.
now.
Let me check. Okay,
Yeah,
but you
maybe.
can go ahead. I'll try to fix
Yeah,
it.
thank you for that. Anyway, congratulations, Aditi, on and a well-rounded presentation. I can say that. Also, yeah, it's clear that you have invested significant time and effort into your research. That's very obvious. And the depth of your analysis really stands out also because you were able to achieve your research objective, or in short, you were able to answer your research questions. Now, my question also is that how did you ensure that your review, remain comprehensive without becoming too overwhelming? Or maybe what strategies did you use to apply, or did you use or apply to keep it focused on the central research question?
So one thing is that like neuroplasticity is a really broad topic, and when I was like going into this, like when I first selected my topic, I didn't want to do it related to like diseases. I wanted to kind of steer away from that. I thought especially with neuroplasticity, I would get a little bit like overwhelmed because together it's like super broad. So when I was like doing my research, a lot of the like I used keywords like neuroplasticity, but I also like when I was picking my two I'm gonna call them like topics like applications in memory and learning, like I specifically only like looked at articles that were completely relating to the memory and learning process. And when I was looking at the stroke rehabilitation, and more than just that like injury, there was actually a lot of like evidence like that could support that. So it wasn't after I like found my two topics, it wasn't too hard to dive into them. But without those two topics, I definitely it would be really hard to like kind of get like confused on how many things there are about it.
Yeah, we're just so proud of you, despite the fact that this is your first time
Hello.
writing a research paper. So, congratulations, Aditi.
Thank you so much. Professor Varkey, is your audio fixed now or?
Yeah. Can you hear me?
Yes, you need to scream like you're screaming at your kids. I don't
No
think you
idea
scream
what.
at your kids enough.
No, I think like that's a different topic. But like, um, I have no idea. Like at my end, somehow I can hear everything clearly, and the audio setting, something is still like, there's some problem. I don't know what it is though. If you're
Yeah.
not able to
Would
hear me
you
clearly.
like to chat the question? I'll go with Sahasra. She's the last
Yeah,
one.
please go ahead. Please go ahead.
Yeah. Okay. Okay, Sahasra, let's go ahead with you. Sorry, you had to wait for such a long time. Everyone, we should have split this. A better planning was needed. I apologize if it took way too much time. We'll do better next time. Okay. So go ahead, Sahasra, and thank you for your patience.
Can you hear me?
Yes.
Okay. Hi, everyone. My name is Sahasra Chikapali, and I go to Psychor Academy, which is a private school located in New Jersey. I decided to do my thesis defense presentation on my research project, which talks about the end-of-life epiphanies, a neurological perspective on memory, perception, and awareness in aging. So let's begin. So to start out, this research explores the phenomenon of end-of-life epiphanies in older adults, and it focuses on whether these experiences are a byproduct of brain deterioration or a distinct cognitive event. So this research shows to study my research question, which is what neurological processes contribute to end-of-life epiphanies, and do they represent a byproduct of brain deterioration or distinct cognitive phenomenons? The topic area I'm investigating is end-of-life epiphanies, specifically in older adults, and exploring the neurological processes that contribute to these experiences. In my project, I had three main objectives. My first one was to examine the neurological mechanisms behind end-of-life epiphanies, especially in regions like the prefrontal cortex and limbic system. My second objective was to investigate the role of brain chemistry changes, such as serotonin and endorphins, in these experiences. My third objective was to determine if end-of-life epiphanies are a byproduct of brain deterioration or a distinct cognitive event. And my last objective was to compare these experiences with near-death appearances, or also NDEs, and explore their emotional significance. So in terms of the literature review, I found that end-of-life epiphanies are often seen in various cultures, involving moments of profound insight or spiritual awakening. Additionally, studies suggest that brain deterioration, especially in the prefrontal cortex and temporal lobes, may trigger the release of suppressed memories and emotions, contributing to these experiences. Additional research suggests that on near-death experiences and altered brain states, like increased gamma wave activity, suggest similarities with end-of-life epiphanies. In terms of my methodology, this methodology for this study involved conducting a detailed survey at a local senior center where participants were asked to share personal experiences or observations regarding end-of-life epiphanies. This survey included both direct questions about individual experiences and inquiries from caregivers about the observations they had made concerning elderly patients. The findings were striking and provided compelling evidence that end-of-life epiphanies are not just rare occurrences, but rather a common phenomenon among the elderly. So in the survey, we found out that 78% of respondents witnessed end-of-life epiphanies in loved ones, 62% experienced profound realization as they age, and 83% of caregivers in the senior center noted vivid memories or newfound clarity in patients. So the significance of this study was that these results indicated that end-of-life epiphanies are common and potentially meaningful experiences, underscoring the need for future investigation. So this survey revealed that end-of-life epiphanies are widespread. Many individuals and caregivers reported profound moments of clarity or emotional resolution before death. A majority of participants observed or experienced epiphanies that involved vivid memories, newfound understanding, and heightened emotional clarity. So this highlights the importance of understanding these experiences as a potentially distinct cognitive phenomenon, not just a byproduct of brain deterioration. So the results of this study suggest that end-of-life epiphanies are common and meaningful experiences observed by both individuals and caregivers, which may serve an important psychological and emotional functions. The findings support the idea that these epiphanies are not simply a byproduct of brain deterioration, but may represent a distinct cognitive phenomenon linked to brain chemistry and memory processing. By examining the neurological mechanisms behind these experiences, this research sheds light on their potential role in the brain's final stages. Ultimately, this study reinforces my research question: what neurological processes contribute to end-of-life epiphanies, and do they represent a byproduct of brain deterioration or a distinct cognitive phenomenon with deeper implications? I contributed to all this research by conducting a survey that provided new qualitative data on the prevalence and emotional impact of end-of-life epiphanies. I also helped connect personal accounts of end-of-life epiphanies to neurological processes, offering a foundation for future research. This research paves the way for future exploration into how these experiences may influence end-of-life care and our understanding of consciousness. Additionally, in this research, I found a few limitations. So the study relied on self-reported data, which may be influenced by memory, biases, or subjective interpretations. Additionally, the survey was limited to a single senior center, which may not represent the broader population of older adults. Lastly, cultural differences in interpreting and experiencing end-of-life epiphanies were not explored, which could affect the results. This is what I also want to explore further and take this research project to a whole new level. Furthermore, I would like to just thank this entire research program, Professor Varkey and Professor Jen, for helping me tremendously throughout this entire process. The research mentors helped me refine my paper, made me understand how to correctly format my information, and helped me with subtopics and areas I could dwell into more. Additionally, Professor Varkey helped me with my paper. His various lessons on different topics were extremely eye-opening. And around his second or third class, when we were talking about diseases in older people, like Alzheimer's, it took me back to a time when I was working at the senior center and was talking to an older patient in her deathbed with dementia. This is when I realized that I definitely wanted to do my research on something related to this field. And this is how I essentially came to pick the research topic that I did. Furthermore, the research program helped me tremendously because before I had no clue how to properly conduct research or honestly what I even wanted to explore further. I was indecisive on what I wanted to study, but being a part of this research program really opened my eyes to not only what I want to research, but also what I want to do in the future as a career. So thank you so much for listening to my presentation.
Okay, Professor Jobin, can you ask her?
That's why I put my question also in the chat in case you are not able to hear. Can you hear me?
Yes, I can hear you.
Okay. So my question to you was first, it's a very interesting topic. When you told that you're going to write on this, I was I had never even read about this myself.
Oh, okay.
So because this is a very unique topic where during the end of life they have very that they have clarity, but why and how it happens in the brain we don't know. So, but since you through your research show that it has got some kind of significance, what kind of deeper implications can you think of?
I'm sorry, could you repeat that?
So it's the same question on the chat.
Okay, you can answer on the chat. Okay.
Can you
Wait, let me read it out. Let me read out the question. Since your research shows that the end-of-life epiphanies is not due to brain damage, what deeper implications can you think of?
Yes, that's a great question. Since it's not related to brain damage, some other limitations I could think of would maybe be, since it's not related to brain damage, the older adults, they could like in the research in the whole entire process when I was doing this, what I noticed was whenever I was asking these older adults the question about their research, of course they were very like interested in it and they really wanted to answer, but some of them I could see them giving, you know, pretense answers and just wanting to answer for the sake of it. But additionally, some other limitations, I can only think of one other limitation, which is that what's it called?
Sorry, not limitations, implications.
Implication.
Oh, okay.
I mean, can you think of why this happens?
I feel like because when older adults, as soon as they're, you know, nearing the end of their life, they look like back at their past memories and they really want to, you know, they want to, they just want to like do something that really like summarizes like everything they've done in their life, like one more little like thing that they could possibly do, one more contribution they could give, or and one more like meaning to their life. I feel like that's why mainly like they would want that these end of life epiphanies happen. I feel like that's the main reason. And that's what I've noticed in a lot of people. I've also noticed that in my grandma when I was talking to her about this. So I feel like that's the best answer.
Okay.
Excellent. This is such an interesting topic, Sahasra.
Thank you
I
so
was
much.
like very, very nice. I mean, I'm enjoying every one of it, and I want to thank you for staying to the end. I'm sorry it ran over, but your energy didn't waver at all. It's a wonderful topic, and I can understand that it's very difficult to gather data from people like that because they're not very coherent or their cognitive abilities are not there completely, but it's very interesting to pursue, right? So
Exactly.
Professor Jen, a question for her.
Yeah, first of all, I want to congratulate you on your outstanding presentation today. Yeah, your energy, you're so active and like you're so determined to really present your paper to us. And the depth of your research and your ability to, you know, to deliver and explain your paper is very impressive also. Now, my question is that since this is another question, another direction that are related to writing process. So, given the nature of a literature review, how did you make sure that your analysis, your analysis was not just a summary of previous research, but also it offered a critical evaluation of the findings?
So what I noticed mainly in previous research was that all the research was more directed to like what these end of life epiphanies were and like kind of like examples of them and things like that. But my research was more towards like whether it was like a cognitive phenomenon or actually caused by, you know, brain disease, like actual brain damage, diseases, things like that. So that's why in the senior center I worked at, I not only like passed out the survey to just like patients in general, I made sure like, you know, some patients like actually had like, you know, brain diseases and things like that. And the other patients were just normal adults in the room to really like summarize my research.
Wow, impressive. Congratulations again,
Thank you so
Sahasra.
much. I really appreciate it.
Okay, excellent. We are at the end of our presentation. Thank you for being here for such a long time. I really, really appreciate it. I want to thank Professor Jobin for joining my crazy idea of mentoring this high school kids. Wouldn't have been possible without him. And Professor Jen Tuban, she's amazing. She's an amazing essay editor and a research creative writer. I want to thank each and every one of you for participating in our program, coming here today and presenting it. And your research doesn't end here. It's just the beginning. And I want to see wonderful things happening from you guys. Okay. So I want to wish you well. Everything is going to be recorded. My team is going to spend a long time in splicing, dicing, putting it on the YouTube. And you will be able to link that in your resume. Okay. So without further ado, I'll let you guys go. Thank you everyone for being here. Got it? Bye now.
Thank you.
Thank you so
Thank
much.
you.
Thank you so much.
Thank you.
Thank
Bye
you.
now.
Bye.
Have a good
Bye,
day.
Ketan. Bye, everyone. Ritika, you stay along. I'm going to let everyone else go. Sorry about that. Thank you, Rose. It's been such a long night for you. Thank you for joining, Rose.
Thank you so much.
Yeah. Thank you, Professor Varkey. Thank you. Yeah. Okay.
Excuse me?
Yeah.
I did not get to present yet.
Oh, shit. I'll have would you be able to, didn't I call you?
You did, but I had issues, so when I came back to join, I was just waiting.
Oh, I'm so sorry. Would you be able to send a recording, honey?
Of myself presenting?
Yes.
Okay, yeah, that's fine.
On Canva. On Canva. I apologize for it.
So would I just record and what email would I send it to?
To me, info@giftedgabber.
Okay.
Okay.
Thank you.
I apologize for the inconvenience.
No problem. Thank you.
Yeah, thank you, Gitanjali. Thank you. Yeah, Ritika, how are you? Sorry about that. That was our final presentation for our neuroscience program. I had like quite a few students and we ran over. Okay, so how can I help you today? Let's get started.
Yeah, so I'm planning on joining the BSMD program as an upcoming 2025 application as a senior. So I'm applying this fall. But I would just, yeah,
Okay,
I
go
would
ahead.
just